Hello, everyone, and welcome back to the second session of the day entitled Computational Approaches in Single-Cell Biology. My co-chair for this session is Dr. Nancy Zhang from UPenn. Before starting, I want to mention that thanks to Arjun and the Keystone staff, we now have a Slack channel. So you can use this to interact. In addition to the discussion forum, you can find the link on the homepage of the meeting. And now it's a great pleasure to introduce our first speaker, my wonderful co-organizer for this meeting, the one and only Arjun Raj, who will tell us about single-cell analysis in cancer. Arjun, stage is yours.
Thank you so much. So I guess, this is normally where I would thank the organizers, but I will take the opportunity to thank my amazing core organizer, Shalev, for doing such a wonderful job putting everything together. So I want to tell you about some work that we've been doing on single-cell analysis in cancer. And we've been working in cancer, but the question that we've been very interested in answering is really some of the same questions that we've been interested in answering for the past, God knows how many years, actually many ways back to Michael Elowitz's original work in 2002, which is, I'll make it sort of operational for you. If you take one cell and grow it out in a dish, so now you have a bunch of, let's say, cancer cells that are all grown out in the dish, are all these cells the same? They all look like gray blobs, but, you can probably anticipate the answer to this question, which is no, and you can use whatever single cell assay you want, of which we've seen many brilliant examples. They will all show variability from cell to cell. Really, though, the question that I think motivated a lot of this at the very beginning was not so much like, are all these cells the same, but do all of these cells do the same thing? And unfortunately, I would say for the field, if you take one cell, grow it out in a dish, and you have all these cells that look pretty much the same, the answer as to whether all these cells do the same thing is generally, yes, they do all the same thing. So, are there really any consequences to these differences? It's been hard to find. And I think part of the reason it's been hard to find them is because well, frankly, I think because we weren't looking at enough cells. And so I put the little asterisk here because that asterisk can be, maybe there's one little cell out down there. Cursor isn't showing up. Anyway, this is a large scale field of view. All these little flecks that you might see in the field of view are actually individual cells, and there are many hundreds of, tens to hundreds of thousands of cells on these dishes. And occasionally you might find a cell that actually does something different from the others. So I'll zoom in on this and show you a little movie of this in action. Here are a bunch of melanoma cells, and I'm going to play this video just to, show you the before picture. So these cells are happily dividing before you add drug. Then we're going to add an anti-melanoma drug to these cells. And what you'll find is that most of these cells end up getting, strung out on drugs. They're like stringy and unhappy and probably aren't going to do very much. But if you are eagle-eyed, you might notice that these two cells here don't look like the others, and these two cells actually started from one cell and even underwent a division. And if I play this video for a little while longer, again, this is with drug applied, you'll see that those two cells keep on dividing and eventually form what is like a drug-resistant colony within these cells, within this field of view, which all begs the question, what was different about this cell to begin with that caused it to behave differently from all the others? And to make a long story pretty short, what we found is that normally people think that when you have a rare cell doing something different, that's an example of a genetic mutation. What we found is that there's non-genetic variability that's strongly associated with this drug-resistant behavior. Each of these dots that you see over here on the left is an individual cell colored by the amount of RNA in them as measured by RNA fish. As you can see to the right, there's one cell that has a very high level, whereas most of them are negative, like that little negative cell on the right. There's one cell that has a ton of mRNA, and it's these rare cells, these like Skittles within the population that express these genes that are the ones that are drug resistant. Now, non-genetic, genetic, what's the difference? One of the key differences is the time scale of reversion. So I actually won't be talking too much about this, but I think it's cool and it's a chance to show a movie. So here's one of these resistance marker genes, NGFR, labeled with GFP. And if you watch it over time, if you watch that cell, you'll see that it goes up and down over time. So it can actually move into the state and then back out of the state. That's part of what makes it a little hard to track down exactly what's going on in these cells because they're constantly flipping back and forth. So what I just showed you is an example of going from distinct molecular states, so different, these cells express these genes, and then linking that to a state in the fate in the future, right? Drug resistance. One of the things that we'd really like to do is be able to start with the phenotype, let's say drug resistant cells, and if we could somehow rewind the video, go back in time, say like, oh, okay, here's an individual, the originating cell, what was different about that cell? So the only way you can do this is to use a cellular time machine, and this is the work of Ben Emmert. I'm just going to go over this pretty quickly because this paper was just published recently. How do you actually make a cellular time machine? So what Ben did, this is a method we call rewind, is you use these lentiviral barcodes that are both in the DNA but also expressed into the RNA. So we barcode all of these cells by transducing with this virus. We then let the cells divide. So now we've got this like these twins, right? So we've got the orange twins, the green twins, the purple twins, the color of the different barcodes. And then what we do is essentially the twin experiment, like separated at birth. So we take these twins, we separate them at birth, and it's not quite at birth, but you fix half of them for downstream analysis and the carbon copy. So that's like freezing them in time. And then the other set of cells, what we'll do is add drug to them and see which ones form resistant colonies. So in this case, purple is the lucky winner. So the purple barcode grew out and made this purple colony. Then what we do is we say, okay, well, now we know in the future purple wins because we sequence all the colonies and we find there's like the purple barcode. And then what we do, it's a little bit of a crazy experiment. We actually make RNA fish probes based on the barcodes that we found by sequencing. And using those probes, we can actually light up the purple cell in this carbon copy. So these are the cells that before they even got drugged. So this is how you go back in time. We're basically going back to this original population and asking, well, can we find the twins of the ones that went on to become resistant? And then ask, what's different about those cells? And we can do all kinds of different profiling. I really won't go into the details of this too much. I'll probably just show you mostly just some imaging because pictures are cool. Okay, so part of the thing that makes this super challenging is that the frequency with which these cells arise is very, very low. So you really have to look at, many, many thousands of cells. The frequency is 1 in several thousand, depending on the day. But I am pleased to report that after a large amount of optimization, Ben was able to find these essentially needle and haystack cells. You can find these very rare barcoded cells in fields of many hundreds of thousands with high sensitivity and specificity. And they express the markers that we've come to know and love. So, AXL, NGFR, EGFR, and a host of other markers that we were able to discover through sequencing that I won't talk about. But basically, you label this barcoded cell and you can find these other markers co-expressed in that same cell. And you can quantify this pretty nicely. So you can, here's again, AXL, EGFR, NGFR, the y-axis is a number of RNA. Most cells on the dish have very low levels and the pre-resistant twins have very high levels. And, there's a lot more to this that I'm not getting into right now in the sake of time. But one of the things that I wanted to focus on was a really clever question question and technique that Ben used in this rewind paper. So one of the things that he did was he asked this question of whether different initial molecular states could be responsible for different outcomes. And to put this in a little context, when you grow enough of these resistant colonies on the dish, what you'll see is that, well, some of these resistant colonies are big and some are small and some are kind of in between. But they're differences. They don't all look the same. So Ben asked this question, well, are the ones that go on to form small colonies the same as the cells that will go on to form the big ones? Is it just, the cell landed in a dish that made it, in the part of the dish that made it small or with certain neighbors that made it big? Or is it all programmed in the initial state of the cell? So what Ben did is he said, okay, well, let's rewind separately the ones that form large colonies from the ones that form small colonies. I won't go through the details of exactly how he could do this, but what he could do then is ask, well, what's different about the cells that went on to form the large ones versus the small ones? And, again, I'm glossing over a lot here, but he could find differences in the initial state of the cell that led to these differences in the ultimate outcome. So what this means is that it's all predetermined. It's all there to begin with. And that determines sort of what you're going to do when you grow up. And this actually really changed how we thought about this problem. Initially, we thought, okay, look, there's some states you become resistant, some states you become dead, right, as a cell. And actually, what his results revealed is that there's at least a potential that there's a lot more rich, subtle behavior going on here. You've got some that are become very resistant and some that seem to become sort of resistant, and who knows what all in between. So this was a question that got Yogesh Goyal, a talented postdoc in the lab, very interested in this. He said, okay, he was just asking the question, how many different resistant types are there? This, very resistant, sort of resistant kind of thing is not particularly systematic or satisfying. So Yogesh did what any modern data scientist would do is, he collected a lot of data, used a neural network to classify all the images, that neural network being his brain, filtering images from his eyeballs, where he just looked in the dish and he just said, hey, look, you can see these different morphological categories of resistant cells. They, some of them seem to kind of grow on top of each other. Some of them look more like fibroblasts. Some are really kind of 3D. Some almost look epithelial. They're these different, and they kept showing up, these like classes of resistant cells. So one of the most important controls that he did early on was ask whether these now seemingly emergent resistant cell types, do they persist through clonal expansion? Essentially, do they breed true? It's actually very much like the old Luria-Delbruck experiments. They had to check these exact same things, whether cells bred, whatever. So the way that he would do this, right, is you would take these cells, you add drug, some cells survive, they grow into little colonies, and then what you do is you isolate and expand. So the way you do that, if anybody's ever done this, is you take this little cylinder and you put it on top of the colony and you put some trypsin in it, and it's all very messy and it leaks, and you're like, oh my God, this sucks. But somehow you can do that, and what you'll end up with are these colonies that have grown out. And what he found is that the phenotype bred true. So if they're kind of swirly looking, then you grow them out and they stay swirly looking as long as you can go. The big problem with this, of course, as I've been alluding to, is this is kind of a fussy thing to do, and many of the cells don't really survive this process, so it's really biased, and we don't really have a sense of what is this like ecosystem of resistant colonies. So what Yogesh did is he developed a new technique that allows us to actually in a more unbiased and high throughput way, figure out what are all the different resistant cell types. So he used actually Ben's same barcode system to do this. So what he did was, it's a slightly different design. He barcodes the cells, adds drug, lets them grow. And then the thing is, every resistant colony comes from a single cell that is now grown out and has a particular barcode in it. And then what we do, so now you have cells that have different barcodes all on the dish. They're all resistant cells, but all the cells in a colony have the same barcode. What he would do is mix all these cells together, put them through the 10x machine, and now you get a transcriptome for every cell, but also you do this little side reaction. I won't get into the details of molecular biology, but you do the side reaction, and what you get are, you can link up the lineage barcode to the cell ID from the 10x, and now what you can do is say, okay, this cell's transcriptome, that had the yellow barcode, and this other cell's transcriptome also had the yellow barcode. This one had the pink barcode, and this one also had the pink barcode. So now we have all of that information. But first, let me just show you what you get straight up out of the 10x if you just do one of these UMAP things. What you get is what looks like kind of a ecosystem of resistant cells. I should say this is kind of wild. Like it's kind of interesting to take a step back and think about it. We started with essentially like a single cell on the dish, grow it out, add drugs. All these cells are as homogeneous as can be. You add drug, and all of a sudden you've revealed all this hidden variability. So these differences are real and have sort of systematic and interesting differences here. So for instance, this one big cluster on the right, these are actually SOX10 positive resistant cells. This is something that shouldn't exist, by the way, based on the conventional literature, and for reasons that I'll get into in just a little bit. You can find these ones that look kind of like neural progenitors. There's some over here that look like smooth muscle cells. Other ones that express, all different kinds of markers. So these are really distinct sort of types of cells as far as we can tell, or at least they have a lot of differences to them. Okay, So we also have all this information about the colonies, right? This is just 10x. This is just all the cells mixed together, not worrying about their colony status. So what Yogesh wanted to know is whether the expression clusters that we find match the colonies that the resistant colonies that we can now identify using the barcodes. So what he could do is paint the barcode. So these are all related cells. You can paint it on the UMAP, and you can see that generally speaking, but in fact, almost all the time, what we see is that barcodes map to specific expression clusters. So these expression clusters are really resistant colonies. So you'll see there's, this one over here has a lot, this smooth muscle colony, right? There's this neural progenitor colony. And over here on the right, what you'll see are a bunch of individual cells out there, and that's why these have not been detected before. They're basically single cells sitting on the dish that nobody would ever bother to isolate and be like, oh, look at this resistant single cell. They'd always focus on the colonies. But there's a lot of these single cells that's sitting on the dish that are actually resistant, and they make up this distinct category of resistant cell. So there's also something that kind of harkens back to these discussions about Waddington landscapes. And I think, Michael was talking about this idea of potential wells and so forth. I actually think that, we're so used to thinking of potential wells as a way of, cells, cell types kind of push themselves into these wells where they're all next to each other. I actually think that's not true. Necessarily, and it's not what's going on here, at least that much I can say. So for instance, if you look over here, this is this VCAM1 positive cluster. Cluster of gene expression, right? Now we have two colonies that both kind of map to that, right? They're kind of both the same resistance subtype. But if you look, they're actually systematically different. They're kind of next to each other on this UMAP. And you see this for essentially all resistant colonies. It's very rare that they actually really fully enmesh with each other. What this is saying is that all colonies are individuals. Some of those differences between those individuals may be small, some of them may be big, but they're each their own they've all found their own path to resistance. By the way, these differences tend to be ECM proteins, so make of that what you will. Using bulk RNA-seq from these colonies, we can link these morphotypes to the expression clusters. So those also match up pretty well. And, these different types of cells are not just they look different. They have very different properties that are relevant for cancer. So for instance, you'll find that they have very different invasiveness. I'm not showing these data, but there's also big differences in proliferation between these different resistant types. And, it's not just some cell culture artifact. If you do these patient-derived xenograft models in mouse, you can find all of these same resistant subtypes appearing in vivo as well. So one of the big conceptual questions that we had, again, is are all of these fates predetermined? So Ben's data definitely suggested that, well, at least, some of these fates are very, rigidly typed from the beginning. But now we could ask this question far more generally. So what Yogesh did is he said, okay, well, I'll do this fate map technique. And what I'll do is I'll just take identical twins and do the 10x on the resistant colonies from both, and then ask the question, do twins go to the same place? And amazingly enough, they do. In fact, they fully interdigitate. So it's unlike the colonies that are sort of next to each other. They all really go to the exact same place. They, almost always go to the exact same place. Even these singletons, those single cells on the dish, they're just because one is a singleton, the twin is highly likely to be a singleton as well, suggesting that, again, these are not just random single cells sitting on the dish. They're doing something, based on what they were before. And just to, make this point clear, what this is saying is that everything is predetermined. Twins always go to the same place. Even colonies that are different from each other but seem to map to the same resistant type, they're slightly different from each other, but the twins always go to the same place. It's almost eerie how much everything is all predetermined in this system. So another thing that Yogesh did, which I thought was really interesting, is okay, well, that's what this ecosystem looks like at one drug dose. But if you add a different dose of drug, you get many more resistant colonies. You get all different sorts of things going on. What happens when you go to a lower dose? And what happens to the to this ecosystem? And in principle, you could imagine many, many different things going on here. So to carry this ecosystem analogy a little further, let's say you had frogs, eagles, and squirrels. So one of the things that could happen is that you have the frog turns into a toad at low dose. So you go to a low dose, and what used to be a frog is now some different component. Another possibility is that you alter the distribution. So some things that were squirrels now become eagles. And yet another possibility is that you get, some wholly new thing coming in entirely, like in this case, bats. And what I can tell you is that all of these things happen when you go from high to low dose. We see examples of all of these. So I'll lead you through a few of these. So one example is the component identity shift. So this is like the frog to toad. So you can see over here at the high dose, we have the UMAP has this big bulge over here. At low dose, that bulge seemingly shifts over to the left. And this is actually this melanin A subcluster. Now, of course, without barcodes, it's hard to say, I have this population, and at high dose, I have this population. At low dose, I don't know necessarily that one turned into the other, but because we have the barcodes, we can actually ask, does the twin that shows up in this region at high dose, where does its twin go at low dose? And you can actually see that those barcodes almost entirely map one region to the other. So this is an example of something that would become a frog at high dose becomes a toad at low dose. We also see many examples of components appearing and disappearing. So if you look here at high dose and low dose, you'll see here are two patches of things that only show up at low dose. Here's one big patch of things that basically only shows up at the high dose. This is actually, also pretty colocalized with this NGFR expression, this, neural progenitor thing. So one of the things so in principle, these could be either, introduction of a new component or loss of a component, or fate switching, that, something that was going to this part goes to somewhere far different. So what we see actually for this blob over here is that it does do this fate switching. So if we look at the twin, so the twin that at high dose goes to this cluster actually switches more generally to this sort of axial positive resistance subtype. So it actually is switching its subtype. This is an example of fate switching. So this would be the squirrel turning into the eagle. But there are other examples where you can see the appearance of new resistant types. So for instance, if you look over here, we've got this patch that is not there at high dose, it is there at low dose. And if you look at the barcodes over here that correspond to low dose, basically you'll find none of those barcodes show up anywhere at high dose. So this is a completely new population. These are basically cells in the dish that when you give them high dose of drug, they don't survive. But at low dose of drug, they do survive and seem to form some independent or new type of resistant cell that's specifically only there at the low dose. Okay, so hopefully I've been able to convince you that there's actually under this seemingly homogeneous set of cells, if you are looking for the right phenotypes and adopt a sort of phenotype first view of single cell biology, I think what we have found is that there's a large amount of this cryptic variability. And once you know where to look, it's seemingly everywhere. And this variability can emerge or sort of revealed by adding different perturbations. And it depends on the perturbation. So you're going to get different outcomes at high dose, low dose. Different cells are getting selected. Different ones undergo different sorts of reprogramming processes, which I didn't really get a chance to talk about today. We've mostly here, I've been talking about everything in the context of drug resistance, but I think these same principles are going to apply across a wide range of biological processes. And we've already been looking at this in stem cell differentiation and IPS reprogramming. And I think we have some interesting results there too that hopefully I can share with you next. With that, I would like to thank all the people in my lab who I'm so lucky to work with an amazingly talented group of people. Ben Emmert really sort of transformed the lab with his barcodes and working out all these details I didn't have time to go over. Yogesh has really taken it in a super interesting direction. I also really want to thank all our various collaborators. In particular, I want to call out Meenhard Herlin and Ashi Veeraratne. As you might have guessed, I'm not really a melanoma biologist, but Meenhard and Ashi most certainly are. Beyond just being, leaders in the melanoma field, they're also just very kind and open-minded people who are willing to listen to kooks like us talk about our single cell, this and that. So, and I want to thank our funders and thank you for your attention. I'll take any questions.
Wonderful talk. Thank you very much, Arjun. We have a question from Siwan Zhang. How much of those differences are driven by genetic variation, mutations, or polymorphism, or rather epigenetic imprinting?
That's a great question. So the variability that we see is, okay, what I can tell you definitively is that the time scales are probably something like 5 to 7 divisions for moving out of a particular, hang on one second, let me get back into the Zoom. The time scales are probably 5 to 7 divisions. Now, that would seemingly preclude things like, SNPs and reversions. I mean, that's like very unlikely to occur on that time scale. Are there genetic things that could occur on that time scale? Sure, like some kind of, episome copy number variation. It's possible. I think it's unlikely for various reasons, including, you don't see like synteny or any of these things that you might expect from sort of large scale chromosomal changes. In a way, it's kind of interesting, right? Like, I think, we're very used to having this need to come up with a molecular mechanism. In a sense, it kind of doesn't matter. The only thing that really matters is the time scale and the reversibility. In terms of what you want to do about it, then yes, I think being able to manipulate it. So we've done CRISPR screens to actually find things that can manipulate these rare cell states. And the very fact that you can manipulate, like we don't find a bunch of like DNA repair mechanisms and pathways that seem to control that. So we do find some, quote unquote epigenetic modulators like a H3K79 methyltransferase. But we also find various signaling proteins and transcription factors. So I would say it's just kind of like run-of-the-mill biology, just hooked up in an interesting way to give rise to this interesting phenotype.
There are many questions, and I'm just going to pick the next one on the list. How do you consolidate this idea of a predetermined difference in the resistant cells with the idea of plasticity between states? Is there some sort of bi, tri, quad stable state that is hard but possible to get out of? And what drives this?
Yeah, so there's a very interesting question also as to whether, I think we're very used to thinking of discrete states, that like you're the gray cell or the green cell, or you're, primed or not primed or whatever. And I think that's actually very misleading. I mean, the results of Yogesh showing that like every colony is its own individual, I think speak to this. Basically, it's a continuum. It's a continuum of states. There's all these different, continuous axes of variation. And somehow that variability can manifest itself in different ways upon perturbation. And it depends on the perturbation, because clearly you're going to end up in different fates depending on which drug or what concentration of drug you give. And that'll either push you one way or the other, So I think it's actually more of a continuum.
Okay, there's a lot of questions, but unfortunately we're running out of time, so I'll just take one more from Matthew Pandelakis. How do you control for the random integration of the lentiviral barcode? Cancer integration has an impact on the drug resistant phenotype.
Sorry, could you say that last bit again? I just
so whether the specific random integration of the lentiviral barcode could affect the drug resistant phenotype, the integration points themselves.
Yeah, it's in principle possible. We don't think so, because you can, like if you just look at like the overall resistance proportion of cells that like had the integration versus didn't have the integration, it's pretty much the same. So we don't think that we're getting like some wholesale change. It is possible that individual barcodes are like going into a region that like changes something about the cell that causes it to become resistant, but we haven't seen evidence for any such singular event.
Do we have any more time for more questions, or is that?
I think unfortunately we need to move on, and we'll continue in the discussion forum or the Slack channel.
Okay. So moving on, it's my absolute pleasure to introduce Stephen Quake from Stanford University, whose work is probably very familiar to many of us at this conference. Without further delay, I will turn over to Steve.
Thank you. It's terrific to be here. Can you all hear me? Yep. Okay, great. So this has been my sort of favorite talk title to use recently. The cell is a bag of RNA. It's meant to be a little bit provocative. And it's also meant to evoke sort of a historical resonance of this battle between biochemists and cell biologists about whether or not you can think about the cell as a bag of enzymes, which is the way the biochemists think about it. And the cell biologists say, no, you have to think about all the complex structures and compartments and so forth. And this is the history of that phrase, bag of enzymes. It dates back to the 1930s or 40s. And you can see the battle has waxed and waned over time. And of course, the cell biologists are right at the sort of some level that those structures really do matter and the cell can't do what it does without them. But on the other hand, biochemistry, by using that simplifying abstraction, has had such a powerful impact on our understanding of how cell works that you can see it's useful to make these sorts of abstractions and then revisit the full complex problem later. And that's been sort of the approach I and many other people at this meeting, I think, have pursued with respect to single cell biology, which is just, let's use these powerful tools we have to look at RNA and enumerate all the RNA molecules in the cell and just imagine it's a bag of RNA and forget about everything else for a little bit and see what we can learn from that. And I'll take you through some examples of that. First ones, I'll just highlight a few recently published things and then get into some unpublished work in the latter half of the talk. So, it's been appreciated for a very long time that even in a phenomenon as complex as memory, that RNA and RNA synthesis plays an important, crucial role. And in fact, these were some very early single-cell transcriptome experiments done by Kendall and collaborators to work through that and discover some of the genes involved. Tom Sudoff and I decided to try to revisit this program using a more complex model, mouse instead of slug, C slug, and which has a sort of more sophisticated form of memory and the powerful tools of single-cell transcriptomics. And we did that using fear aversion training. The mouse is put in a cage and a tone is played and gets a shock and it learns to associate that sound with something unpleasant. And then you can play the sound and the mouse freezes because he or she recalls the unpleasant behavior, consequence, stimulus. And we've used the very powerful TRAP2 mouse developed by Leach and Low to try to encode which neurons are being activated when the mouse recalls its memory well after the stimulus. And when I say we, I mean two very talented postdocs from our labs, Shan Jiang in Tom's lab and Michelle Chen in my lab. And so we're doing the recall more than 2 weeks after the memory was formed. So this is really long-term memory formation and recall. And the cells that are involved in the recall are made permanently fluorescent. We then analyze them by single-cell transcriptomics. And sure enough, we're able to find very clear differentially expressed genes and sustained transcriptional signatures well after the event. As you sort of dig through those genes and pathways and classes, ontology classes, you find, for example, very clear view of vesicle exocytosis, which is upregulated in those cells, and as well as many other interesting effects that I don't have time to do justice to here, but wanted to sort of flag this as one example of how one uses this approach in conjunction with all the other powerful tools of cell biology to look at a really complex behavior. As another example, and here just a single slide on this paper, this is work done between my lab and Leach and Low's lab and a bunch of other folks involved as well, but an attempt to use single-cell transcriptomics to understand the evolution of anatomical structures in the brain, in this case, the cerebellar nuclei, which are part of the cerebellum. And we did comparative studies in chicken, mouse, and human, and found that it had been known that these structures are present in different numbers in these species, in invertebrates. And we were able to show that these evolved by having conserved cell types and duplicating those over time. And this is, I think, an example of what's going to become a very productive research area in the coming years of using these single cell transcriptomic atlases to understand evolution at the level of cell types and the role of cell types in evolution. And I think there's a great future in that. So now, let me tell you, so those are things that sort of done in my lab as regular science projects between collaborations between two groups. Let me tell you now about some of the larger projects we've embarked on using the resources of the BioHub, which have involved many different labs across institutions to do things that are sort of what would be difficult to do in any one lab or project or grant. And so I began exploring the idea of establishing organism-wide cell atlases and thinking about them as references that would be companions to the genome that will help you understand how genes are used in various cell types. And we began in the mouse. We called it the tabula muris, and then the tabula muris senis, the mouse aging cell atlas. And this is work that was led by myself and Tony Wiscaray, and at the BioHub involves Spiros Damanis and Angela Pisco, and many, many other groups at Stanford and UCSF and the BioHub, and I'll acknowledge some of them as we go on. And here we looked at something like 18 different tissues, looked at roughly 500,000 cells over them, and looked at them at various ages of the mouse, including out to 30 months, which is like a human centenarian. And part of what we did here was use a very small number of mice because we took all the tissues from the same mouse, and we worked very hard on the coordination so that these essentially the tissue-to-tissue comparisons were perfectly controlled for age, epigenetics, environmental effects, and so forth, because they're all taken from the same mouse. And that became a theme of how we've approached these atlases. And so to do that, it involved, as I said, a lot of coordination. This group of, we involved tissue experts, labs that were experts in each of the tissues we were studying. The student or postdoc from that lab all showed up, ice bucket in the hand every morning. A mouse was sacrificed, and they all took their organs back to their labs, did the dissociations and the sorting, brought them back to the BioHub, where all the cDNA was made and the sequencing was done. And the leaders of the sort of BioHub platforms there were Norma Neff on genomics and Jim Carcanius in data science. And it was, as I said, a lot of coordination, but a lot of fun to do. It's all published, so I'll just, two slides on the data here. One is the ability to look at aging-related effects in shared cell types or shared cell states across tissues, in this case, senescent cells, able to see that number of senescent cells is increasing with time, and the expression of key genes involved in senescence, like CDK2A, is increasing as well. And more generally, as a tool to kind of, these single cell atlases are a tool to deconvolve effects that get mixed up in bulk measurements. Like, is it the number of cells expressing the gene that's changing, or is it the level of expression of the gene that's changing? And we can, with single cell approaches, can separate those two effects, and have done that more generally in this work. Also, we've been able to look at not just gene expression, but because some of the data was taken with SMART-Seq2, able to have whole transcript sequencing and to look at mutational load and how that changed with age, and look at that in comparison with RNA spike-ins. The third paper in this series of mouse atlases is not published yet, but it's available in bioRxiv. And this is a functional study where Tony and Tom Rando spent years kind of exploring this, have stitched together the vasculature of two mice. That's called parabiosis, when two mice share the same blood supply because their vasculature is attached. And if you do that with mice that are of different ages, it's been observed that the old mouse starts to appear younger, and the young mouse starts to appear older. So there's a rejuvenation for the old mouse and accelerated aging for the younger one due to this shared blood supply. And the reasons for it have been unknown, and we decided to try to understand some of the molecular basis of this by building an atlas of these parabiosed mice. And so we did it again with a large number of tissues, and because we have the reference of natural aging, we can now compare accelerated aging with natural aging and ask, is that similar? And we can compare rejuvenation to aging and say, are things really getting reversed? And we can do that at the level of many different cell types across tissues and number of different genes that are differentially expressed with aging. This is like a master table that kind of shows it. The little scale is the key is at the bottom. The things on the left side of the circle are the accelerated aging. On the right side are rejuvenation. And blue is consistent with regular aging. Red is not consistent. You can see many of these differential genes, especially the ones that are downregulated with age, are consistently reproduced both with rejuvenation and accelerated aging. Whereas upregulation, it's a little more mixed and not as clearly consistent with natural aging when you have this parabios model. But to give you one sort of bottom line story here, as you might expect, energy matters here, and genes involved in electron transport mitochondria are predominantly downregulated in age across many, but not all cell types, but the vast majority they are. And these are ones that are well reproduced, both with the accelerated aging, and you see it going the other way with the rejuvenation. And so there are some broad themes that are unfolding from this. And again, I'll refer you to the preprint if you'd like more details. And in the remaining bit, I'll walk you through some of the other large-scale efforts we've undertaken collaboratively to build these reference atlases. We're approached by the folks in the Drosophila community who were looking for some assistance in organizing an effort to do a fly atlas, and we've been part of that. And this involves a number of different labs around the world. So the overall coordination is Leach and Lowe, Norbert Perrimon, David Sutherland, Stein Ertz, Bart DePlanck, and myself, and Hongji Lee, who just was a postdoc in Leachin's lab and just became independent faculty member at Baylor. And we've involved, again, tissue experts to do tissue by tissue analysis of the flies. It involves 13 labs from all over. We've done most of the sequencing at the BioHub, some at the NIH, and the data processing and analysis has been led by Stein and Bart's groups, and they've been great about bringing in the larger community to do annotation jamborees. And so, again, like our mouse data, a huge number of these cells have been hand annotated by experts, and people have really thought about what the cell types are in the context of existing cell biology knowledge, and thereby provide a lot of sort of added content. This is one example of one tissue body wall, thorax and abdomen. And you can see that cell types that had previously been identified by just 1 or 2 or 3 genes now have entire transcriptomes associated with them and allow a much deeper understanding of what's going on. We've got, this is an older one, this is 250,000, a little higher than that now, pushing 400,000, I think now, cells with some form of annotation. And a couple hundred different cell types that have been really carefully looked at. And we think this is going to be a great resource for the fly community going forward. Data is all taken, papers being written, also analysis is being done. So you can look forward to this appearing on bioRxiv and having the data released sometime in the near future. Another one we've been working on, and which is close to completion, is a non-human primate, the mouse lemur. Lemurs are very interesting primates found in Madagascar. The mouse lemur is on the evolutionary scale almost exactly in between mouse and human, the primate, but it's very small, and it has many properties of a model organism like the mouse does that make it attractive to work with. And my colleague at Stanford, Mark Krasnow, has spent many years trying to develop it as a model organism, and he was our lung expert on our mouse cell atlas, and convinced us that we should use the tissue expert teams to also do a lemur cell atlas, because he has a colony at Stanford, and the lemurs occasionally have to be sacrificed due to old age. The vets say it's time to put them down at some point, and that happened a few times recently, and so we pulled a group together to build a lemur cell atlas. About 250,000 cells, 30 different tissues, in the first non-human primate cell atlases. And again, there's been an enormous amount of effort put into hand annotating the cell types. And again, paper is almost fully written, and so you should look forward to seeing this appear again in the not too distant future. We're very excited about it. It's been an enormous amount of effort put into and trying to understand the various cell types and connect them to what we know about human and mouse cell types, returning to this theme of looking at evolution. And there's some very interesting cell types that appear that the lemur shares with human but not with mouse, a couple in the lung, for example, as well as other tissues. And then finally, we're working on a human project as well, following the same sort of philosophy where we're trying to obtain something like 20 to 25 tissues from the same individual donor, so that you can look at shared cell types and have it perfectly controlled for genetic differences, environment, age, so forth and so on. And this is led by Bob Jones in my lab, and involves, again, a very large number of people across all three institutions, 3 universities in the Bay Area, as well as the BioHub. We're just kind of at the beginning of this. We've done a few practice. We've been working closely with one of the organ procurement operations that finds donors for transplant, and have been doing some practice attempts. And we've done a pilot with 6 organs, a pilot with 18 organs from the same individual. We just did one with 20, and that data is on the sequencer right now. We've done many of the organs individually, so from different donors, but we've sort of got it all kind of coordinated and working now, and we're pretty excited about what this will serve as a resource going forward. And I'll just sort of close by saying, as I've tried to emphasize, these are really team efforts involving large numbers of people, faculty, students, postdocs, staff members. These are pictures of some of the folks who are involved in mouse and lemur, and with human, it's an even bigger list. These are many of the tissue experts involved. In the picture, you see some of the core team members who are there pulling all-nighters when the donor's available. It's kind of a real complicated dance, and it's something because we're working with the intact cells and not the nuclei, we're doing it all in a pretty narrow time window, and have got that working pretty well right now, and thanks to the efforts of these folks. So I'll stop here, and happy to take any questions, and just return to this notion that this abstraction of thinking about the cell as a bag of RNA is a very powerful tool for understanding cellular identity, and we think these large-scale atlases are going to be super powerful companions to the genome and understanding how different cell types and tissues use the genome. Thank you.
Thanks very much, Steve, for an excellent talk. I was happy to see the small and test time on the new atlases. I was using them in the Tabula Moris, so that's great.
Terrific.
So we have a few questions. One from Zhu, from the young age parabiosis dataset, could you tell if certain tissue ages rejuvenate earlier than the others?
Yes, there's very interesting data on this kind of time course effect, and of aging, and the strength of the effect. And I'll refer you to the preprint there to have a look at it. And not all rejuvenate. It's not uniform, right? It's definitely effect size changes. Sorry, go ahead. The next question.
I'll ask a different question. Very impressive atlases. This is from Homaira Hamidzada. In these large atlases, how do you go about ensuring robust reproducibility? Besides pooling several tissues, would it be feasible to reproduce in a different lab and assess the stability of gene signatures for each cell type population?
Yeah, that's a very good question. So, of course, we're doing biological replicates, and that's one way to assess reproducibility. Another way is, we've been kind of concerned about the systematic errors involved in the measurement. And so I alluded to this briefly, but I'll expand on it now. We've tended to use 2 different technical approaches to do the transcriptomes. And so, for example, with Tabula, with all of them, well, no, not with all of them, but with the mouse, for example, we've used both 10x and SmartSeq2 to obtain transcriptome data, and we can try to understand what are the kind of technical artifacts that each of those methods introduces by comparing the data sets from both of those and looking what's shared and what's not.
Thanks. We have another question from John Murray. Any plans for developmental atlas generation in interesting comparative organisms like mouse lemur?
I'm sorry, what was the question?
So whether you're planning to do a developmental atlas in
developmental
organisms
atlas.
like mouse lemur?
Yeah. Not with the lemur, because we can't, ethically sacrifice them at young ages. There we're being opportunistic, and we're only working with them when the vets say it's time to put them down. But with the mouse, obviously we have, we also with the fly have an aging series as well. That's coming after the first fly atlas, but we're working on a fly aging series also. And obviously, there's other folks who have worked on early development, like fetal and those sorts of things. And, the Sanger, among others, have been working on that in mouse and human. And we're kind of leaving that to them to do, because they're doing a fine job. And we're focusing on what happens after birth.
Okay, great. I think we'll take one last question from Carmen Gerlach. In your parabiosis experiment, did you establish the atlas by taking into account whether the RNA comes from cells of the older or younger partner, having either stayed in the original host or crossed over to the partner, using congenic markers or alike?
Yeah, that's a very interesting question. So they share the blood, but not the solid tissues. And so, most of the analysis focuses on the solid tissues. So we're fairly confident that they're, which mouse they come from. But obviously there's ambiguity there with any cells of the immune system in the blood, which come from either. And in this case, they weren't separately labeled.
Okay, thank you very much. So we'll be moving on now to Nancy Zhang from the University of Pennsylvania. Nancy, the floor is yours.
Okay. Thank you, Shalak, and thank you also to Arjun, for both of you for organizing this wonderful conference. I will tell you about work that we have done on exploring intratumor heterogeneity through the lens of allele-specific copy number and chromatin accessibility. So, as we all know, cancer is a disease caused by the evolutionary expansion of cells in the body's tissue microenvironment. We now know that this evolutionary process is driven by genome instability and also epigenetic plasticity. And the degree of plasticity and the degree of genome instability fundamentally shapes the course of each individual disease. So to really understand how tumor clones evolve, we need to tease apart the contributions of DNA level mutations and epigenome level changes. And in this talk, we're going to focus on two dimensions of these complex entities. We're going to be looking at DNA copy number, and we're going to be looking at chromatin accessibility. Okay, so just, I think this is very familiar to most people, but just to bring everybody to the same page, copy number alterations refers to gains and losses of large chunks of our genome. And most copy number analyses you do see now in single cell studies are limited to the quantification of total copy number, which should be 2 in normal diploid cells. But we're going to look at allele-specific copy number, and you will see why in just a bit. For allele-specific copy number, you need to look at heterozygous SNP size, as shown here. And so, for example, in a 1-copy amplification, the total copy number increases to 3, but we want to know that it is a blue haplotype that got amplified. And then, so the variant allele ratio is 2 to 1, or 2 out of 3 for the blue haplotype, and the red haplotype is at 1 out of 3. And then for deletions, you have a loss in total copy number, and the variant allele frequency also changes from the 50% that you would expect in diploid cells to 1 and 0. And then that is a loss of heterozygosity, which sometimes you can also achieve without a change in total copy number. So, for example, gene conversion events create situations like this, where you have a segment of the genome where the variant allele frequency is at 1 and 0, but the total copy is still at 2. So it's actually been, I think, exactly 10 years since the first single-cell DNA sequencing study of cancer was published, which gave a very detailed portrait of the evolution of a breast tumor in terms of DNA copy number events. And since then, there has been quite a lot of technological innovations for single-cell DNA sequencing, as well as computational innovations for methods to analyze such data. Now, I want to point out that most of these methods are limited to total copy number, with the exception of CHISEL, which considers allele-specific copy number. And let's talk about that more next. So the reason why we are interested in allele-specific copy number is because we believe there's a lot of hidden heterogeneity in the tumor that you don't see if you don't look at things in the allele-specific manner. So I thought this was a very interesting study 4 years ago that came from, I think, the Tracer X consortia in the UK, where they looked at lung cancer. And they did multi-region bulk sequencing. So bulk sequencing by taking multiple slices of the tumor. And what they found was that for a region of amplification, at different regions of the tumor, different haplotypes were amplified. So there would be regions where the blue haplotype was amplified, and there would be regions where the red haplotype is amplified. And this aspect of intratumor heterogeneity is not visible if you only consider total copy number, or if you look at allelic ratios, but average across the entire tumor, as you see on the left, right? So they're all at 50% if you average it. Now, we wonder how prevalent such events are and what is their contribution to tumor evolution. So just a month ago, this very nice paper came out in Nature Biotech for this method called CHISEL. Now, CHISEL is the first method that looks at allele-specific copy number for single cells. This is a very hard problem, and I will tell you why it is hard in just a bit. But CHISEL, because of the sparsity of single cell data, it is applicable only to very high coverage single cell DNA sequencing data. And because it's looking at allele-specific copy number, it relies on external phasing of these heterozygote sites in order to get reliable pooling across adjacent SNPs. But nevertheless, they were able to identify some instances of such mirrored subclones in their analysis of a breast cancer sample that was sequenced at 10x. So now I'm going to actually switch gears a bit and talk about single-cell ATAC-seq data, because we're going to be looking at both single-cell DNA and single-cell ATAC. And single-cell ATAC is now widely adopted for cancer studies. But we know it is an obvious fact that the accessibility of a genome region depends on its underlying copy number in the given cell. And so this variation that you see oftentimes in UMAP plots, so usually what you do is you do a UMAP of the peak coverages, and then you get rid of the immune cells, or you separate out the immune cells from the tumor cells. And for the tumor cells, you can see that there's oftentimes within intratumor variation in the chromatin profiles. But how much of this is actually due to the copy number differences between these cells? So it's hard to tease this apart. What existing studies do is they do these smoothing averages using very large windows across the genome. And then for each large window, they would normalize the coverage in a tumor cell to a normal control. And it's not clear what to use for that normal control. So what is often done is people will look at, what other cells are sequenced, like these, endothelial cells and fibroblast cells, and then rely on them as a control. But that's really not a great idea, because even when you look at very, very large windows, almost even chromosome level, there are broad shifts in coverage that have nothing to do with the underlying copy number. For example, just a simple experiment. If you take endothelial cells, which, so these endothelial cells in this sample, and then normalize them in this way against fibroblasts, you will see that there are these windows, these regions at the scale of entire chromosomes or chromosome arms that are elevated or deleted, that look deleted. Okay, and then same is true if you look at fibroblast cells and use endothelial cells as control. It's just the reverse of this, right? So it's not clear what to use as a control, and therefore we can rely on these kind of analyses to do like a bird's eye view kind of sanity check, but it's very hard to do a confident high resolution analysis. So another reason why we want to look at allelic ratios is because we hope that by adding allelic ratios to the mix, we can get more confident copy number estimates for single cell ATAC-seq data. So, but why is this such a hard problem, and why are there, currently so few methods to do this? I'm going to try to sort of give you a setup of the problem. So, consider here this genome region, this genome region containing these 9 SNPs. Okay, and our cell population contains multiple subpopulations, right, with different copy number configurations. So the top one I showed you is normal. Then this middle one, clone 1, has what I just put in, a loss of heterozygosity, copy neutral loss of heterozygosity here on the right. And then clone 2 has an amplification of this segment. Okay, so this is the underlying structure that you want to recover, but you don't see. What you do see is for each cell, your data is, you have many cells, and for each cell, you see a very sparse sampling of reads. And they may be clustered in peaks if you were doing ATAC-seq, or they may be scattered somewhat randomly if you're doing single-cell ATAC-seq. So if a read by luck overlaps one of these SNPs, you will know which haplotype it comes from, right? But usually you don't have that many reads that overlap with these SNPs. So cell 2 is more realistic, right? So I showed a lot of overlap just to show you the concept. So the challenge is that in single cell data, both the reads are sparse to begin with, and those that overlap with heterozygous sites are even fewer. And so when you're doing allele-specific copy number, you need to sort of gain power by pooling across adjacent SNPs, but you don't know the phase, so you don't know which alleles to sum up, right? You don't know that this A, B, and B are actually part of the same haplotype, so you don't know how to sum them up. And furthermore, you don't start out knowing where the breakpoints are. Okay, so I'm going to show you, for the rest of the talk, I'm going to show you how we address this problem. I'm going to describe to you the alleloscope, which is a method we developed to estimate precise allele-specific copy numbers for single-cell DNA-seq data, as well as for single-cell ATAC-seq data, which are usually much lower coverage. And for these, we require that there be a matched bulk DNA-seq control. Okay, and our method, unlike CHISO, does not require external phasing information, and it can achieve higher accuracy under low coverage. And I will show you that using this alleloscope, we can see much more heterogeneity among tumor cells than we used to. And this also allows us to dissect the contributions of chromosomal instability versus chromatin remodeling to driving the intratumor heterogeneity. And I'm going to skim over many technical details in the interest of time. If you want the full picture with rigorous benchmarks, you can look at this bioRxiv preprint that we have up. Okay, so just quickly going over the pipeline. Alleloscope starts out with input matrices that are the allele-specific counts at heterozygous sites. Okay, 2 matrices. And if you were doing single-cell DNA-seq, you can just aggregate across these matrices to get a pseudo-bulk, and then perform segmentation on the pseudo-bulk via joint modeling of the total coverage and variant allele frequency profiles. If you were doing single-cell ATAC-seq, we actually recommend doing the segmentation on the paired bulk DNA sequencing data. Okay, now we were reluctant to do this, to just say, okay, let's get the bulk and do segmentation on the bulk. But what we found out is that even if you were to try to do find a smart way to sort of segment the single cells, you wouldn't be able to recover those rare events as much as if you just do a bulk. So that the concern is what if rare events get smoothed out, right? But if you use a low enough segmentation threshold, you can actually, catch them, and then it's okay because we're going to process them next. So for each segment that you get out of the segmentation, we take the heterozygous SNPs in the region, and we perform we have a statistical model where we perform a likelihood-based expectation maximization procedure, where we iteratively infer the phase of the SNPs by pulling information across cells, and then using those phasing to infer a cell's major haplotype proportion by pulling across the SNPs using that estimated phase information. And then we're going to iterate this over and over again. So what we exactly mean by major haplotype proportion, that's a very key concept. So major haplotype proportion for a given cell is a proportion of the haplotype that is dominant in the cell population. So if, for example, for this cell, if a pink haplotype has, is dominant in that it's a majority in the population, then the major haplotype proportion is 2 out of 3. Okay, or if sometimes it doesn't have to be that the dominant haplotype in the cell is dominant in the population. It may be that this gray haplotype is actually present at a higher proportion in the population, in which case the major haplotype proportion will be 1/3. Okay, so we're going to estimate these major haplotype proportions, which are key quantities that we carry forward in the next step. Okay, so we're going to move over here, and then we're going to take this major haplotype proportion across regions and then use that in an algorithm, which I'm not going to describe, to identify the normal cells as well as the diploid regions. The reason why we want to do this is we want to use them to get a relative coverage. Okay, so because it's hard to normalize the coverage in single-cell ATAC as well as single-cell DNA, you've got to have good controls. So we're going to find these good controls to get a good normalized coverage, which gives us now for each cell in each region a pair of values, the row hats, the coverage, and the data hats, which are the major haplotype proportions. So we're going to plot them against each other in this, what we call a phase diagram. Okay, and then in this diagram, you can see that there are these canonical points where we expect the points to lie if we did things right, okay? So for example, if the cell is diploid, then we expect the major haplotype proportion to be 0.5 and rho to be 1, okay? Because there's no gain or loss. If there is a copy neutral loss of heterozygosity, then we expect it to be at this blue point here, okay? Where rho is 1, theta is 1. Now, losses are here, okay, at the purple point. And then these points here are the amplifications, okay? 1 copy amplification, 2 copy amplification, okay? So there's actually, 5 canonical points for 2 copy amplification. So if there's one thing I want you to take away from this slide, it is this diagram with the canonical points representing integer allele-specific copy number ratios. Okay, so let's apply this alleloscope to a cancer and see what we get. Okay, so this is the very first sample we looked at is a primary gastric tumor. Okay, and you can see from the cell ranger, this is cell ranger plot from 10x, where you have basically the total copy number estimate. You see that this is not a very complex tumor in the sense that there's not a lot of events. I mean, we see much more complex, messed up genomes in cancer than this, right? And that's expected because this is of MSI subtype. Okay, so what happens when we submit some of these regions to alleloscope? So let's first look at this chromosome 7 amplification here. If you look at the chromosome 7 amplification under alleloscope, you do expect this cluster here that represents the normal cells at 1.5. And you do see this, what is expected is amplification at 1.5, 2/3. But what surprised us is that we immediately found this mirrored subclone at 1.5, 1/3. So, wow. So because we were thinking, this is literally the first region and first sample we looked at, and we landed on a mirrored subclone. And at first we thought this must have been an artifact, a single cell sequencing artifact. But let's look at, so these are the mirror subclones. Let's look at this chromosome 8 event. Okay, so you see this normal cluster at 1.5. And then we do see this expected 1.5, 2/3 one copy amplification here. But look at these small tight clusters on top of these canonical points. What are they? Are they the outliers? If they were outliers, we don't expect them to fall exactly on the canonical points, right? So, by our model, these should be the other allele-specific configurations of amplifications. And you don't really see them really in the 10x cell ranger plot. And we were surprised that, what seems to be a pretty genome, chromosome stable tumor, actually the first two events we looked at, they were so complex. And just for the sake of it, let's look at a deletion here. You do see the deletion cluster, but we see these 2 points down here that seems to mirror that event. So this genome is loaded with these very interesting complex regions. Okay, are they real, right? So we were actually, we didn't want to jump to conclusions because we know that there are lots of different kinds of artifacts in single cell sequencing. Lots of things can go wrong, for example, dropouts and amplification biases. So are these complex mirrored events real, right? So we developed a validation strategy using bulk linked resequencing, which sort of leverages microfluidics to partition and barcode high molecular weight DNA to give us haplotype phasing. So we can get for the same sample, for these samples, we get the phases. And then so we can use these phases to assess how well alleloscope is doing in terms of phasing the SNPs. And we can also plug in the linked read phases to the second stage of alleloscope to get the estimated genotypes, right? And compare whether the genotypes are equivalent. So it turns out that all of our events are validated by linked reads, and we get 100% genotyping accuracy on this sample. Now, if you look at other samples, we looked at quite a number of samples, and we see that indeed we get a lot of complex regions. I mean, this seems to be the norm rather than a special example. So, for example, we see cases where you have a gradient of haplotypes across cells for this amplification in the population. And I just want to point out that 2 of these samples are metastasis. So we do see that even in metastasized cell populations, we have a lot of hidden genome instability. So if you look at single cell ATAC-seq data, so this is a public data set. So we did the segmentation using bulk whole exome sequencing. And if you look at the clusters, they do lie on the canonical points, but because the coverage is much lower, the dispersion is high, right? So, but nevertheless, we are able to find this subclone delineating event on chromosome 4. And if we plot the cells by UMAP, by the peak signals, which is sort of the alternate dimension, we see that oftentimes the separation in peak signals is somewhat driven by this differences in this copy number, this LOH. Okay, so now let's look at a really complex gastric cancer cell line by both single-cell DNA-seq and single-cell ATAC-seq. So here first, the single-cell DNA-seq. Okay, so here is the bulk profile. So you see that it's quite messed up. And if you look at some of the regions by single-cell DNA-seq, again, we see these really complex, much more complex than you see using total copy number. And then if you look at single-cell ATAC-seq, you have much more dispersed clusters, but sort of centered on the same canonical points. So, it does correlate. And then if you take the estimated allele-specific copy number profile for single cells and then do a heat map, you can discover very, very interesting clonal heterogeneity. And then remember, for ATAC-seq, we also have the peak profile, right, which is sort of orthogonal to this because, they are sort of much more local. And then if we do a UMAP embedding of the peak profile and then color the points using the clone memberships, we see that most of that difference, that variation is driven by this underlying clonal membership. And then what's more is that because we have the clonal membership of the cells, we can now compare the peak signals across clones and look for differentially accessible peaks after accounting for the differences in their underlying the copy number of the region, so that we can actually get a better identify a sort of more interesting peaks that are still significant after adjusting for copy number. And I believe these are the ones that we should follow up on if we want to look for interesting chromatin remodeling events. So there's a lot more to say about this, but I just want to stop here and give a summary. So, we developed this method, alleloscope, which we think will be useful for single-cell DNA and ATAC cancer studies. And through this, we found, I think, previously sort of not understudied pervasive occurrence of highly complex multi-allelic copy number aberrations. And this allows us to interrogate the interplay between chromosomal instability and epigenetic plasticity in tumor evolution. And so I want to acknowledge all the people who did very hard work for this. Most of what I talked about is due to the efforts of one very talented student, PhD student, Qiming Wu at Penn. And also I want to acknowledge Zilu Zhou and Yu Chaojiang, whose previous work led up to this, and our collaborators at Stanford in Dr. Hani Ji's lab, who did all the sequencing experiments and discussions that led up to this study. And thank you for listening.
Thanks a lot, Nancy, for this elegant talk. We've got a few questions. What do you make, this is from Joshua Tyson, what do you make of the small number of cells that fall between 1.5 and 1.5.67 on your plot? What is the biological meaning of these scores?
The small number of cells? It's probably rare subclones.
The region of 1.5 and 1.5.67.
Yeah. So, yeah, some of these subclones are really small. That's what you mean, right? They are really small. But at least for
the first few plots that you showed, he's asking about the. Okay, maybe we can continue this one on the discussion. Okay, so can alleloscope work for only the single-cell ATAC-seq? If yes, how the performance is comparing to with bug DNA-seq?
The single-cell ATAC-seq, you get more out of it because you have single-cell level data and you can look at allele-specific single-cell copy number, right? I mean, I think in the least, yes, these clusters are very, very noisy. These, they're still very noisy compared to the DNA counterparts. But when you combine information across regions, you do get enough signal to assign these cells confidently into subclones. And that's not something that you can do using bulk DNA-seq.
Okay, thanks. I have a small question about the first part of inferring copy number variation from the single-cell RNA-seq. So you showed this nice endothelial fibroblast example. I'm just wondering if there are computational tools that might be more robust, for example, looking only at the housekeeping genes, because I guess these are sort of endothelial, highly expressed genes that are just by chance located on some chromosomal region. That's why they're higher in endothelial cells compared to fibroblasts and vice versa. So is there any sort of computational work around this to get the copy number variation from RNA-seq?
Yeah, I think that's a good idea, but people haven't done it. And I think, harnessing housekeeping genes might be a good idea. For ATAC-seq, it's not clear which regions we should be using as housekeeping regions.
Okay, great. Julia Wang actually also asked, can single-cell RNA-seq data be leveraged for the detection of allele-specific copy number variation? We would love to do that. We get asked that question, and so can this method be applied to single-cell RNA-seq as well? It's more complicated because in single-cell RNA-seq, you also have the transcriptional stochasticity at play there, and that introduces more noise. You see that right now, at least right now, the limit of our method is to apply to single-cell ATAC-seq. We can, I think there needs to be a lot more work done to get this to work for single-cell RNA-seq. Okay, let's take one more question from Laura Su. Have you looked at tissues that can undergo rapid expansion? Do you see changes in allelic copy number under physiological conditions?
We have not, and that would be something we would be interested in doing. Just with this method, I think we would want to apply it to such cases.
Wonderful. Thank you very much, Nancy. We're going to go out for a coffee break now, and we'll reconvene in 20 minutes. So thank you all.
Hi, everyone. Welcome back. And we have 4 more very exciting talks to round off this section. So our first speaker is Jean Fan from the Department of Biomedical Engineering at Johns Hopkins. Jean will talk about modeling and visualizing RNA velocity. Go ahead.
Okay, well, thank you, Nancy, and thanks to Shalev and Arjan for the opportunity to share some of our work here today. Among this rather star-studded lineup of speakers and leaders in the field, I might add. Although we're not exactly enjoying the slopes as we initially intended, I think when we got the invitation a few years ago, I definitely do hope that everyone is staying healthy and safe from the comforts of wherever you're joining us from. So let's see. Right, before I jump into things, I just wanted to first give a shout out to my team members. We are a reasonably new lab starting in the midst of the pandemic a little over half a year ago now. So we're definitely still recruiting and looking to establish new collaborations, particularly with more experimental labs. So if you're interested, please do reach out and we can get a conversation started. So I think it's well appreciated by this particular audience and highlighted by a number of talks recently or already, for example, by Michael, Itai, Shalev, Long, and a number of other speakers already, that the advances in sequencing and imaging technologies have recently really enabled this high throughput spatially resolved transcriptomic profiling at single cell and near single cell resolutions in fixed tissues. And here I'm just highlighting a few different technologies on the tissues that they've been applied to. And of course, there's many more, but it's hard to fit them all into the same slide. But briefly, these technologies allow us to measure not only what genes are expressed within these cells, but also preserve how these cells are organized within the tissue. So of course, given this very large and high dimensional data, we need, for example, new computational and statistical approaches to analyze them and extract out the relevant biological insights. So for more on our thoughts regarding some of these computational challenges in particular, and in particular, I guess, as we apply these tools and technologies to disease settings, please do check out our review in Nature Experimental and Molecular Medicine. Well, one of these technologies pioneered in Xiaowei Zhuang's group, where I conducted my postdoc research, is called multiplexed error-robust fluorescence in situ hybridization, or MERFISH. And what MERFISH does is it uses combinatorial labeling, error-robust barcoding, and sequential imaging to achieve spatially resolved genome-wide now transcriptome imaging with a very high, near 80% detection efficiency and a very low 4% error rate. But, given this very high detection efficiency and low error rate, it allows us to begin asking some interesting biological questions. So in particular, in our PNAS paper from 2019, we applied MERFISH to image 10,000 different, unique gene species and began asking questions related to the enrichment of different genes and different subcellular compartments, for example, exploring how mRNAs are organized within the cells. So we can, for example, combine MERFISH with ER staining and ask, what genes are spatially co-localized on a pixel level with the endoplasmic reticulum, and essentially test on a statistical level what genes are enriched in the ER versus outside the ER. And being that this is an imaging-based approach, it's also very nice that we can just essentially visualize the results. So we can visualize here, each red point is a ER-enriched gene species that we identified, overlaid onto the ER staining image. And visually, we can just begin appreciating how the different ER-enriched genes are indeed spatially co-localized with the rough ER, even in the very fine mesh networks of the rough ER here. And likewise, we can, of course, check individual genes and so forth. But zooming out, we can also, for example, look at cellular organization and ask, for cells of a certain transcriptional state, if we look among its K nearest spatial neighbors, do we see any enrichment of particular cell types and cell states? And in this particular case, we were looking at, U2OS cultured cells that comprised 5 different transcriptionally distinct cell states. And indeed, cells in cell state 1 tended to be spatially co-localized with other cells in cell state 1. And again, we can just visually appreciate this in this plot here, where each point is a single cell, and they've been colored based on their transcriptionally distinct clusters. And visually, we can see how cells of similar transcriptional states tend to cluster together with each other in culture. But of course, we can also apply these technologies to other cell types and even tissues. So more recently, this is work led by Guiping Wang, a very talented graduate student who is now pursuing a postdoc in Howard Chan's group. But she applied MERFISH to image and characterize the spatial organization of, I believe, over 4,000 different genes within cultured hippocampal neurons. And what this allows us to do is just begin really visually interrogating and also statistically interrogating the different spatial organization of different RNAs in, for example, the dendrites or more distal dendrites within the axons and other possible subdendritic and subcellular compartments of these highly polarized cell types. For more information on this, I do encourage you to check out her preprint that's available on bioRxiv. And of course, this data is very large, and so just exploring it can become a bit challenging. So we actually built on advances in video game graphics and rendering to develop an interactive web tool that allows us to explore, these millions of different mRNAs and their spatial organization. In this case, this is the neuron data set again, where, for example, each point is a different mRNA, and they've been colored based on species. So just visually, you can begin appreciating how, we have GAT2 and some inhibitory neurons here, and CAMP-K2A is more spatially co-localized to the distal dendrites, whereas KIF2C in blue is more co-localized with the axons, for example. But the story I really want to teach you all about today is actually in regards to a particular limitation of these approaches, and it's that these cells are fixed. So, they're dead. There's no way for us to follow them as they do, interesting things. And ultimately, the expression measurements that we obtain represent static snapshots in time. So even though, these measurements represent static snapshots, it doesn't mean that all temporal information is lost. And we can see that concept here where we have, the static snapshot of friends jumping off a cliff. But we're all smart enough to be able to infer in this particular case a pseudotemporal ordering regarding which friend must have jumped off the cliff first. And actually, with a few additional assumptions, such as how gravity goes down, we can actually provide a direction to the pseudotemporal ordering. Now we know that this friend must have jumped off before this friend, and so forth. And actually, given the information regarding the positions of these friends along this dynamic process, we can actually also begin predicting the future positional states of the people on top of the cliff at a future time point. They must be more like the next friend. Even though gravity goes down, we don't expect them to just plummet downwards. There's some information regarding angular momentum that we can infer based on the spatial positioning, in this case, of all the other friends. So this is kind of the same intuition behind RNA velocity analysis, where we can take advantage of the spatial patterns, in this case, in this static snapshot to make inferences regarding the future transcriptional states of these cells. And in particular, for RNA velocity in situ analysis, we'll leverage the nuclear and cytoplasmic expression levels to predict the future transcriptional state of these cells. Under these very simple assumptions that, if we recall from Biology 101, that mRNAs are transcribed in the nucleus, which we can observe from our static snapshot, and they're exported into the cytoplasm, which again, we can quantify and observe for each gene and each cell in our population. And presumably these mRNAs are eventually, translated into proteins and degraded and so forth. But that means, given this very simple model, well, we can model the cytoplasmic mRNA expression level, or the rate of change of the cytoplasmic mRNA expression level, as a function of what's coming in minus what's going out. And this means that at a certain steady state where, this rate of change is 0, we would expect there to be a certain ratio of the nuclear and cytoplasmic expression levels for a particular gene. And that means that when we look across a population of cells, if we, for example, see that some cell has more nuclear expression than what we'd expect in steady state, then we can interpret this cell as upregulating this gene, since there's more nuclear expression, there's more nascent transcription, and so forth. And alternatively, if a cell has lower nuclear expression relative to cytoplasmic expression compared to what we expect in steady state, then we would interpret this as this cell downregulating this gene. And indeed, when we look at real data, again, x-axis is cytoplasmic expression and y-axis is nuclear expression for these particular genes, MCM6 in this example. We can see some cells, with higher nuclear expression and some cells with lower nuclear expression relative to cytoplasmic expression. And likewise for this different gene, KIF2C. So it seems that our model is appropriate for our data, and our data likewise adheres to some of the assumptions or the patterns that we expect to see under our model. So what this means is that, for example, for a particular cell, we have this high dimensional observed transcriptional state. In this case, for a particular cell, we have some genes that are lowly expressed in blue and some genes that are highly expressed in red. But in addition to this observed transcriptional state, we can then use our RNA velocity models to predict the future transcriptional state of this cell. So for example, perhaps, here there's a gene in the middle that's kind of lowly expressed in the cell, but perhaps this cell, has higher nuclear expression relative to cytoplasmic expression. So in the future, we predict this gene to become more upregulated and turn red, for example. So what this means, of course, is that we can still do all our clustering analysis. We can take, for example, our matrix of genes by cells, and we can do our clustering analysis to project this data down to 2 dimensions using two-stochastic neighbor embedding or other graph-based embedding and dimensionality reduction approaches. And we can also identify transcriptionally distinct clusters of cells. So this is actually U2OS cultured cells, where, again, each point here is a cell, and the cells are closer to each other in Disney space if they're more similar transcriptionally, and they've been colored based on the graph-based clustering. So there are approximately 5 transcriptionally distinct clusters here. And again, we can do, differential expression analysis and find that MCM6 is most upregulated in these C2 cells, and KIF2C is most upregulated in this C5 cells, and also visualize the expression of these genes in the Disney plots, with red being high expression and blue being low expression. But of course, in addition to all this, as we would typically do, what RNA velocity analysis, as I noted previously, is because we have both the observed state and now we can predict the future transcriptional state, we can actually also project that predicted future transcriptional state onto this 2D embedding as well. And in this particular case, we can represent this predicted future movement in this lower dimensional transcriptional or lower dimensional embedding of arrows. And what we can just appreciate is that in this particular case, it goes in a circle. And perhaps if you're familiar with some of the genes that I mentioned previously, it's unsurprising that these would actually reflect the progression of these cells through the cell cycle. So we can actually take the angle along this circle here as the putative pseudotime and begin looking at the expression of different genes as a function of pseudotime itself. With, for example, MCM6, as we saw previously, was upregulated and then it becomes downregulated, versus KIF2C has a bit of an opposite pattern. But we can actually do this. We can then, look across all the genes that we've evaluated and test, on a statistical level, what genes have a significant proportion of their variance, for example, that can be explained by pseudotime, and in this manner identify 1,000 different genes with transcriptional variants that seems to be associated with the cell cycle, so that we can, in this particular visualization, begin appreciating the gradual transcriptional changes that the cell has to go through in order to, as it progresses through the cell cycle. And of course, we can also apply these RNA velocity approaches to more complex systems, such as, in this case, neurodevelopment. And just visually, we can, in a very unbiased manner, for example, identify, in this case, the neuroprogenitor subpopulations that's giving rise to the different glial populations, and alternatively, differentiating into the neurons, as well as begin teasing apart some of the transcriptional dynamics that are occurring at these, early cell fate decisions, for example. So, however, the approaches that I've described so far for visualizing RNA velocities have relied on projecting, these observed and predicted future transcriptional states onto a 2D embedding that's actually made from the observed transcriptional state alone. So this can present challenges, particularly as we begin applying these approaches to more complex cellular trajectories, such as, for example, here we have a single-cell RNA sequencing data of pancreas differentiation, which is a system which is reasonably well characterized, and we expect there to be, for example, cycling ductal cells that give rise to endocrine progenitors that then give rise to the four terminally differentiated hormone-secreting cell types of the pancreas. So we can take the single-cell RNA-seq data and, again, do different types of dimensionality reductions, such as PCA, Tisney, UMAP, diffusion maps, and so forth, and again, overlay our velocity arrows onto these 2D embeddings. But depending on which embedding we choose, we get slightly different, or at least we capture slightly different aspects of the underlying cellular dynamics. In particular, for example, maybe Tisney and Tisney and UMAP better capture the branching trajectory of the more terminally differentiated cell states, and also the cycling ductal progenitors, which is a feature we don't necessarily see, for example, in PCA. So, given this conundrum, what Lila Atta, a talented MD PhD student in my group, sought to do was actually create a 2D embedding directly from the RNA velocity itself. And she developed a software package called VeloViz to make this method accessible. And for more details of the method, you're definitely welcome to check out our preprint, which is on bioRxiv, as well as access some the software and tutorials on our lab website. But briefly, what this method does is it takes advantage of the current and predicted future transcriptional states from RNA velocity, and it computes this composite distance for all cell pairs that takes into consideration not only the transcriptional similarity between the predicted future transcriptional states of each cell and the observed cells in the population, but also the direction of that RNA velocity. Great. And then what this allows us to do is create a higher dimensional graph representation of each cell with every other cell, taking into consideration, these predicted future transcriptional states. And then we can, of course, prune certain edges of this graph based on other, high spreading distances or similarities. And of course, then visualize the resulting high-dimensional graph in 2D using various graph-based embedding approaches. And when we apply this VeloViz approach to pancreas development, again, what we see is that we can recapitulate the expected cycling ductal cells that differentiating into the endocrine progenitors, and then eventually give rise to the terminally differentiated hormone-secreting cell types, mirroring our expected underlying cellular trajectory. However, so far, the cellular trajectories I've shown you assume a reasonably uniform sampling of cells along this dynamic process. But what happens when intermediate states are missing? And this is a challenge that we faced in some of our other collaborations, particularly in the disease setting, where we were concerned about certain cell populations being lost due to sensitivities and dissociation or other biological and technical challenges. And it's of concern to us because what we find is that a lot of these 2D embedding approaches that use only the observed information are able to capture, for example, these local relationships, in this case between friends, but it's not necessarily very good at bridging these gaps. And we can just simulate an example of this, for example, by removing, in this case, the pre-endocrine progenitors from this pancreas development single-cell RNA-seq data set. So we can remove, the pre-endocrine cell here and rerun all the velocity projections and 2D embedding. And expectedly, what we find, perhaps most best highlighted in this UMAP, is that even though these 2D embeddings are able to capture the local trends, in this case, capturing the cycling ductal cells and the progression into the EP cells, it doesn't necessarily preserve the global relationship across this particular simulated gap. And in this particular case, we might be led to an incorrect interpretation of the underlying biology, where we might naively assume that the endocrine progenitors are differentiating into something over here, and they're not at all connected to the terminally differentiated homosecreting cell types, for example. In contrast, in contrast with Velovis, because we are actually taking advantage and into consideration the predicted future transcriptional states, we are then able to essentially bridge this gap and connect these endocrine progenitors into towards these hormone-secreting terminally differentiated cell types and create a trajectory that's more consistent with the known underlying biology in spite of this simulated gap. And of course, we can do this also not only with the pancreas, but we can go back to the MERFISH U2OS cycling cells that I showed previously and just show that without Velovis, we can still recover this very nice circle of cells progressing through the cell cycle. But then we can also begin introducing a simulated gap, for example, by removing all the G2M cells and rerunning all this analysis and redoing the embeddings. And in this particular case, Velovis is able to still keep this cycling trajectory and relationship among the cells, but now, of course, we have a little gap where the G2M cells used to be before we removed them. Great. So then I hope I've been able to show you that with single cell and spatially resolved transcriptomic data, we really do need new computational and statistical tools to extract out relevant biological insights, as I'm sure many of the speakers in this lineup agree with me. But data from single cell and spatially resolved transcriptomic profiling technologies do represent, at least for now, static snapshots in time. So we can overcome this by using, to some extent, with computational modeling via RNA velocity analysis to predict the future transcriptional state of cells. And I hope I've been able to show you that by taking into consideration of these predicted future transcriptional states, we can create more stable representations of the underlying cellular trajectories. And with that, you're welcome to check out some of the tutorials and resources we have on our lab website if you're interested in trying out these analyses for yourself, or even just reproducing some of the results we have in our papers. You're definitely welcome to check out our lab's website at jeff.works. And with that, thanks to everyone again for your attention. This is my lab right now. This is our virtual lab holiday card this year since we weren't able to reconvene together. But with that, I'd also like to thank, in particular, my postdoc mentor, Xiaowei Zhuang, and some of the grad students, Chenlong Xia and George Emanuel in the lab, who generated the MERFISH data, as well as my PhD mentor, Peter Karchenko, and Ruslan Sodatov in the lab for his efforts in leading the original RNA velocity analysis. With that, please do feel free to reach out, and you can also find me now and then on Twitter.
That's great. Thanks. Thanks, Jane, for the wonderful talk. I'm just going to go down and the list of questions. The first question from Jared Sosberg is, are you able to make any inferences about spatial rather than transcriptomic trajectories of cells, i.e. in a system such as the intestinal epithelium where cells undergo unknown translocation?
That is absolutely a really exciting question, and definitely something that we're looking into currently, whether we can not only predict the future transcriptional state of cells, but let's say take advantage of cells at different, migratory paths or, spatial locations within a dynamic migratory process, and see if we can try to predict a potential migratory path to the observed cells. So definitely a really exciting direction, and one that we're looking into and interested in collaborating with others.
Thanks, Jean. I want to ask my own question here. So you showed very nicely that you infer velocities not from intronic, non-spliced and spliced versions of the RNA, but rather from nuclear versus cytoplasmic levels. Can you comment on the advantages, disadvantages, and can you also use MERFISH by imaging intronic probes and exonic probes to get similar velocities?
Yeah, it's a great question. I guess for context for everyone else's benefit, the original RNA velocity analysis pioneered in Karchenko lab with Stella Narson's group was based on single-cell RNA sequencing, where we leveraged the ratios of spliced and unspliced RNAs under the assumption that, RNAs are transcribed in an unspliced state, and then they get spliced and as they mature and get turned into protein. So it's the same logic where we can look at spliced and unspliced RNAs to look for, predict whether certain genes are being nascently transcribed and upregulated. Now the short answer is, in theory, yes, we should be able to I guess if we had designed specific probes for, the introns of RNAs, and we could distinguish that from the fully spliced RNAs, perhaps by, using probes that span the exon junctions, I think it will be possible to do an imaging-based version of, the spliced and unspliced quantification. I do think that the nuclear and cytoplasmic, if we have imaging data, nuclear and cytoplasmic might be a more robust way of quantifying, these dynamics compared to spliced versus unspliced. For, I guess, a number of reasons related to how difficult it is to probe the dynamic range of unspliced RNAs by imaging. That's, I would expect them, as we've seen from Wang Kai's lab's work, a lot of the nascent transcription happens at these small foci on the DNA, so it's hard to tell whether it's one gene or 10 genes, or it's all kind of just spatially aggregated there. It is interesting, though, and an ongoing project in the lab is actually to compare these two models and try to see if there's certain dynamics predicted from one that are not well reflected on the other. And likewise, as we move into disease systems, I think it's really interesting to see if, for example, RNA velocity predictions from in the disease setting might be, for example, more susceptible to alternative splicing or aberrant alternative splicing, but be a little bit more robust to the nuclear and cytoplasmic ratio quantifications, for example.
Thanks a lot, Jean. There are more questions, but we'll have to continue this on the discussion forum or the Slack channel. We need to move on now. So it's a pleasure to introduce the next speaker, Cole Trapnell from the Department of Genomic Sciences in the University of Washington, who'll tell us about studying developmental robustness at whole embryo scale and single-cell resolution. Welcome, Cole.
Thanks very much. I'm really grateful to the organizers for giving me a chance to tell you about work in the lab towards studying robustness in zebrafish development, which is an incredibly robust process. So, early in development, as the cells are sweeping out the lineage and making fate decisions, they're proliferating and growing exponentially. So you can just do a thought experiment where you can ask, Okay, suppose that a particular progenitor cell of some clade there's a little bit more of those in a given embryo. What happens to its descendants? Do you end up with a massive excess of those descendants in the final animal? Because if you did, you'd expect to see, major anatomic defects relatively often. And yet rarely do we see, a zebrafish, a fish swimming around with one little fin and one giant fin. That really only happens in Finding Nemo. In fact, embryonic development is incredibly robust. You can remove 30% of the cells in embryos that are 4 hours post-fertilization, and at 26 hours, those embryos will look basically normal, but they'll be a bit smaller. So we are interested generally in how genomes encode these incredibly robust programs of gene regulation and development. It's widely believed that regulatory genes are organized into circuits that canalize cells into fate decisions and into fates in the proper stoichiometry and proportions. These circuits and signaling pathways regulate organ size, and they allow cells to communicate with one another to self-organize into precise patterns during tissue morphogenesis. So generally, we are interested in knowing which tissues and cell types develop most reproducibly across individual embryos, which genes ensure that reproducible development, and how those genes are organized into circuits and signaling pathways that stabilize the output of the overall developmental program. And we want to be able to address these questions looking at every cell type in the animal and every gene in the genome. And so our tool for this is single-cell RNA-seq, which is great because you can make atlases of development like these 3 that I'm showing you here from the zebrafish. You can find all the cell types. You can count their abundance in embryos. And you can see whether or not they're lost or gained in response to perturbations. And we are interested in knowing how each cell type varies in abundance across embryos and whether or not perturbations alter that variability. But in order to answer that question, we need to be able to look at many, many, many embryos, many replicates, if you will, of the developmental program. And doing that with single-cell RNA-seq is technically and economically challenging. So we've been working on a way to do this, and it's built around a technology we developed last year with Jay Shinduri's lab called Skyplex, in which, and the way we're using it in this project is we're taking an individual embryo, many individual embryos, and putting each one into a different well, and then dissociating the cells of each embryo into little cell suspensions, and then labeling them with DNA oligos that carry in them an embryo identifying barcode sequence. And then when we pull the nuclei out of those cells and pool them together and sequence them all as a pool, we capture not only the transcriptome of each nucleus, but also the oligo that it was labeled with, so we can tell which embryo that nucleus came from. And in order to do this, we need to be able to sequence, lots of cells. So we're using skyRNA-seq, which is a combinatorial indexing-based method we worked on with Jay a few years ago that can allow you to profile millions of cells in a single experiment. So having done that, we can just count how many cells there are of each type in each embryo and compare these counts across time or in response to perturbations. So to get a sense of the baseline variability in each cell type in the embryo, we first created a time-resolved reference atlas of zebrafish development. And so we've sequenced a little bit less than 1 million cells from 859 different embryos, and these were collected at one of 15 time points from 18 to 96 hours post-fertilization. So this window opens when segmentation is ongoing and semitogenesis is happening, and goes all the way through embryogenesis and into the early larval stage. We can tell based on looking at marker genes which tissues are represented, and we can see that the cells come from 33 different tissues and are one of 85 major cell types. So up until this point, this looks like a sort of a usual cell atlas, but the distinguishing feature is that we know which embryo each cell came from, and we can tell that we're sampling about 1,000 cells per embryo. It's not completely even over the time course because we're devoting more sequencing to certain time points than others, but that gives you a sense for how much we're sampling from each individual. Now, how are the genes in the genome, the regulatory genes, organized and regulating one another in order to stabilize the output of the developmental program? So to address this question, we're looking not only at the wild type embryos that I just showed you, but also many mutant embryos. And we're looking at embryos that are, have any one of these genotypes where these lineage, regulatory factors that I'm showing you are knocked out. We're targeting genes that are important in the mesoderm and the central nervous system, and then a few in the neural crest as well. Most of these are transcription factors, but we've got a couple of morphogens and the receptors in there too. And we're doing this with CRISPR. We're doing this by taking advantage of some recent advances in efficient CRISPR mutagenesis, where we're targeting each of those genes with 3 independent guide RNAs and then co-injecting the guides along with RNPs into one-cell stage embryos. And what this allows us to do is make F0s, sort of at scale. We can do a lot of these in a day, and then sequence the F0s with Skyplex. And another advantage is that, in contrast to conventional mutants for at least some of these backgrounds, is that a very high proportion of the F0s have the expected phenotype. So this is a scalable approach. The one downside, which is important to keep in mind, is that we have to accept some amount of mosaicism in these embryos, and that'll be important later. But we have, as controls for that, we've got for a bunch of these genotypes, conventional nulls as well. Okay, so now I'm showing you cells from the mutants, and this is 3 million additional cells collected from each of 812, or one of 812 embryos. Those embryos are from one of the 22 different genetic backgrounds I showed you a moment ago. And if this data looks exactly the same as the previous data set, that's just because we've projected these mutant data onto the reference time series using an algorithm very similar to the azimuth algorithm. Okay, so I'm not going to be able to show you more than just a little peek at the data, but I want to tell you about this vignette from the neural crest. So the neural crest is neural crest cells are a transient and migratory cell population that that some refer to as the 4th germ layer, because these cells migrate out in development all over the embryo and give rise to a bunch of different cell types, as you can see here. And we're targeting 2 transcription factors in this experiment, TFAP2A and FOXD3, both of which are transcription factors important in neural crest cells. And loss of function mutants, single mutants for either of these genes, lead to depletions in pigment cells and other cell types that emanate from the neural crest. And in the double mutant, there's basically no neural crest output. The cells have no pigment, the embryos have no pigment cells, etc. So these are the single cell data for the double mutant. I'm coloring each cell type by whether or not there are more or less cells in the double than there are in control embryos at matched time points. And what you can see is that, as you'd expect from looking under the microscope, there's sharp depletions in the clusters corresponding to melanocytes and xanthophores and the pigment progenitor cells. And there's also a reduction in craniofacial cartilage, which is consistent with this jaw phenotype that the double has. You also see changes in cell types that are harder to spot under the microscope. So here's another view of the same data. I'm just going to show you box plots now of the number of cells of each type across the embryos. And in contrast to controls, you can see that there's really not a reduction in pigment progenitors in TFAP2A single mutants, whereas in embryos lacking FOXD3, they're sharply reduced. And if you look at the differentiated pigment cells like melanocytes, they're altered in the single mutants and completely ablated in the doubles. And this is true for the other 2 pigment cell types as well. We see changes in glia and actually increases in glioblasts, which is interesting. And so we're excited to follow up on that. It's a little bit challenging to make inferences about this population, though, because glia come from the neural crest, but also from other places. We do see the expected reduction in sensory neurons. And so overall, the phenotype that we arrive at by counting the number of cells per embryo for each of these types is totally consistent with what we'd expect, although there are some new observations too, such as we see a major increase in spinal cord neurons. And that's cool because spinal cord neurons are not generated by the neural crest. So we'll have to figure out what's going on there. And to do that, we're going to have to look much earlier than 18 hours post-fertilization. Okay, so you may have noticed, in addition to changes in the mean, there appear to be some changes in the variance in the number of cells per embryo of some of those types. So how do we assess whether that's real? Well, if we want to test whether or not a perturbation significantly alters the variance in the number of cells of a type, we have to confront and address this issue that's very common in count data, which is that the mean of a thing that you're counting, like a cell type, is related to the variance of the thing that you're counting. And so, for example, in this cartoon, each dot is a cell type, and the horizontal axis encodes the average number of cells of that type per embryo. The y-axis is the coefficient of variation, the standard deviation over the mean. So you can see this trend here with your eye, and we can capture it with methods from, say, bulk RNA-seq analysis. We can fit a generalized linear model relating the mean to the coefficient of variation, which we can model as a gamma distribution. And then we can also describe the error in where each cell type is in this plot by fitting a beta binomial distribution to each cell type's counts across embryos. And that gives us a little confidence interval for each cell type. So now we need only ask, if we want to know, is a particular cell type more variable than one would expect for, the typical cell type of that abundance. We just need to ask whether or not it's above the curve enough to escape its own confidence interval. Likewise, we can ask whether or not perturbations elevate the variance enough to be unexpected. And perturbations often change the mean too, and we can account for how much the variance typically changes when the mean changes. And sometimes what you'll see is that a cell type is reduced in abundance and its variability is kind of brought back in line with expectation. Okay, so with this sort of model in place, we can now color our box plots by how variable the cell types are across embryos in each genetic background. And in doing that, we can see that although the average number of pigment progenitors in TFAP2A mutants is basically the same as controls, those cells are more variable. We can also see this trend much more prominently when we look at melanocytes. And overall, I think we can say that in addition to loss of TFAP2A or FOXD3 changing the average output of the neural crest, losing those genes makes it more variable. All right, you can look at covariances too. You can here I'm showing you that melanocytes are positively correlated with xanthophores in wild-type embryos, which is not surprising because they are siblings in the lineage. But if you look at TFAP2A mutants, that correlation is inverted. And that suggests to me, and this is speculation, but it's cool, that suggests to me that we've somehow changed the canalization of this particular fate decision across embryos. One of the other awesome things about zebrafish as a model for development is that they develop externally, so you can perturb them through the environment. And if you raise zebrafish at the reference temperature of 28.5 degrees, you usually get fish that are like straight as an arrow, like this. But if you dial up the temperature even a little bit and still within the physiological range, the range that they get in their natural habitat, you start to see with some frequency embryos with bends in their tails. And if you look closely, they have other anatomic defects as well. So we wanted to understand the cellular and molecular basis of this anatomic variability. And towards that, we've sequenced the transcriptomes of another 700,000 or so cells from 288 different embryos. And these are all wild type embryos, but some of them were raised in hotter water. Now, we want to study the basis of the variability, but in order to do that, I need to tell you about another thing that happens when you turn up the temperature, which is that development goes faster. So if you raise embryos, if you look at a 24-hour-old embryo that is in 28-degree water, it'll look like this drawing on the left. But if you were to do that at 34 degrees instead, the embryo will only take 18 hours. We'll only need 18 hours to look like that. So that's a 6-hour acceleration. That's a lot. And we know we need to sort of account for this somehow when we're analyzing variability in response to temperature stress. And the way we've done that is we've created a matrix where the rows are cell types and the columns have the abundances of those cell types in each of the individual embryos. And then we just feed this matrix into a trajectory analysis. So you just put this thing into UMAP and you get the picture like I'm showing on the right, where the dots are embryos and they're following this linear trajectory through the space of cell frequencies according to time. But what's cool is you can sort of facet the data and look at the embryos from any one time point, and you'll see that the embryos that came from hot water are always a little ahead of the curve. And that's true of whatever time point you look at. So we can capture this acceleration in the cell frequency data. And we know from, like Chuck Kimmel's classic paper on how to stage fish using the microscope, we know how much acceleration to expect at any given temperature. We know we have a quantitative model for that. So we can compare that expectation with the acceleration we see in the cell frequency data, and the two methods agree very nicely, which means that we can stage our fish just with the cell frequencies alone and match them up when we're looking at how they vary in response to temperature stress. Now, it's true that dialing up the temperature accelerates development, but that's not to say that all the cell types are affected equally. Some cell types are more accelerated than others in their development, and we learned this by looking at the transcriptomes of cells from hot water embryos, and comparing them to transcriptionally similar cells from embryos in reference temperature water, and then just looking at the difference in actual age. So when we do that for each cell type, we can see that some cell types, like the notochord, which is an incredibly important patterning cell type, it really helps set up the anterior posterior axis of the embryo. It's much farther along than other cell types, even in stage matched embryos. So not only does dialing up the temperature accelerate development, it introduces asynchrony in development, and that in turn could drive variability across individuals. So which cell types are more variable at higher temperatures? Well, to find that out, we turn to our mean versus variance plots here and picked out a few that are unexpectedly variable and even more so at higher temperatures. And what we can see, like a good example is the pigment progenitors. And what we can see when we look at these guys is, I'm showing you just the number of pigment progenitor cells per embryo spread out over developmental stage on the x-axis. What you can see is that there's not really a big change in the average number of pigment progenitor cells as you dial up the temperature, but there is a big dilation in the variability, and it's particularly pronounced, sort of on the earlier end of development. So you can kind of see that a little bit more clearly in this little heat stripe here. Now, those are pigment progenitors, which give rise to 3 different pigmented cell types. So what happens with that excess variance? Is that like transmitted down the lineage? Are the differentiated cell types more variable as well? Well, it looks like they are. If you look at the 4 cell types, you can see that at elevated temperatures, all 3 pigmented cell types have excess variance at that same stage in development than at reference temperature. And it's to varying extents. Melanophores are more affected than xanthophores, for example. But interestingly, in all 4 cell types, it appears to be kind of tapered off or damped down as the embryos develop further. Okay, so I've only given you a glimpse of this totally huge data set, but I think we can draw a few conclusions. One is that I told you about how we've used Skyplex to sequence around 5 million cells from more than 2,000 individual embryos, many of which had been perturbed in various genetic or environmental ways. Overall, this is a way that you can accurately phenotype or stage individual embryos based on the cell type frequencies. You can do that, and you can also detect shifts in the abundance of each cell type in the embryo, and also sometimes see changes in the variability in those cell types across individuals. We can see that losing lineage determining factors leads to losses and gains of individual cell types, and sometimes increases the variance in the output of a lineage, like neural crest in the case of TFAP2A and FOXD3. Temperature stress accelerates embryonic development of many, but not all, cell types, and that introduces asynchrony, which in turn could contribute to variability across individuals. And it also increases variance in the relative abundances of a number of cell types. So this is what we've learned so far. This is definitely an ongoing project, but I'm very excited about the future. And because this is a computational session, I thought I would highlight what it means for our lab, what this kind of experiment means for our lab, and what new problems we might be able to work on. So, I didn't really talk about this at all, but we have many different genetic backgrounds in this first experiment, and they often phenocopy each other in terms of the cell types that are gained and lost. So I'm hopeful that we'll be able to look at these phenocopying relationships and systematically and automatically maybe assemble the genes that we're targeting into interaction networks, which would be very important for and helpful for understanding how fate decisions are being made and how development is being stabilized. I just showed you like 5 million cells worth of single cell RNA-seq, and I don't think I showed you the expression of a single gene in the whole talk. But many of these methods, I think, could be readily applied to look at variability in transcript levels rather than cell counts. And if we did that, we might learn something about transcriptional regulation within the cells in each cell type in the whole embryo. And I did tell you that there's a strong covariance structure and that you can mess with it in these data with genetic experiments. I think that this could be a rich source of information for understanding the lineal relationships between the various types of cells in the embryo. So these are cool problems in what I think of as like statistical developmental biology, and we're going to be working on them in my lab over the next, I don't know, while. It's going to take us a while. These are hard problems. So I want to conclude by thanking Lauren Saunders, Mike Doherty, and Sanjay Srivatsan, who did these experiments and have been analyzing them together with Maddie Duran, and we've been supported in our team by Brent Ewing, who's a software engineer. None of this would have been possible without the assistance and interpretation and design guidance, and in some cases, hands of 3 other labs, Cecilia Moens, David Kimmelman, and Dave Rabel, who are experts in many of the genes and tissues and cell types that we're targeting here. I want to thank members of their labs and ours. I also want to thank Nola Klemfos from the BBI and Frank Steemers from Illumina for sequencing support in this project, which was funded by the Paul Allen Frontiers Group and the NIH. And I'd like to thank you for your attention, and I'll take any questions if there's time.
Thank you very much, Cole, for this amazing talk. We have a question from Arjun Raj. Very cool. What do you think? Does heat or stress induce variability or reveal cryptic variability?
Well, that is a terrific question. I'll just give you a couple of things that we're looking at to get some more mechanistic insights as to what might be going on. We are very interested in the overall sort of thermostability of the proteome. And we know that different cell types in the embryo have as their kind of repertoire more or less thermostable proteomes, and we're sort of trying to correlate that with how much, with which ones are more or less variable. But we also can see that temperature changes the rate of proliferation. We think it changes the rate of proliferation in certain cell types and maybe not others. So, it's hard to say whether or not this is variability that's being that would be cryptic otherwise. But I think that there's going to be many possible mechanisms, and a number of them will be true, and they won't necessarily be mutually exclusive, if that makes sense. I know this is a subject, Arjun, close to your heart. We love your review on this area from a couple of years ago, so I figured you might be into this kind of thing.
Cole, the next question from John Murray. Really impressive work. Can you see any cases in your mutants where the cell frequency differences are accompanied by observable differences in trajectories, suggesting novel differentiation paths?
So that's a really good question. And by the way, great to, sorry, I'm not actually looking at you, John, when you're asking this question, but it's great to hear from you. So I was going to show this. We can see a clear case. I just didn't have time. We have a bunch of mutants that target muscle development, and you can see very clearly that some of the mutants for like downstream sort of mid-range effectors cause like sort of slowdowns in muscle development. Cells kind of get blocked halfway through the trajectory, and then if you look at their upstream regulators. Or if you look at genes which kind of collaborate with those, you look at doubles, for example, the cells are like blocked completely at the entry point to the trajectory. So you can see, barriers very, very easily, and it's a super high, you could see the effect size from space. We have, I'm not sure that we've seen like cells going completely off in a new direction yet. That's something that we want to do, but one of the more generally, and this gets at a comment that Arjun made in his talk, this discretization that we're doing into cell types has some pros and cons. One of them is that, it makes things simple and you can use all kinds of statistical tools, treating things categorically that it's hard to do in the same way with a continuous picture of the data. But on the other hand, it makes it more challenging to do analyses like I think the ones that you're suggesting. So we're taking kind of a first pass through the analysis where everything is in discrete cell types. And then I think in places we'll want to zoom in and take a more nuanced view where something like that might become more evident.
Thanks, Paul. There's time for one more question, so I'll just combine two methodological questions. Dihu asks, why are you using siRNA-seq instead of 10x? And Chris McGinnis asks whether it would be possible to use just a more concise set of 100 lineage identifying genes instead of whole transcriptome sequencing. Because you're interested in tabulating the cell types rather than cell space.
That's a great question. So I'll address the first one first. I mean, the most basic answer is that SkyRNA-seq is like much, much cheaper than 10x. Just the library prep is just much, much cheaper, especially because, we're doing it it's very DIY, right? It's in-house. So that's one thing. And we need to be doing millions of cells in order to be able to reach the statistical power that we need for these kinds of analyses. So that's just a kind of a power and economics issue. The other thing is that our hashing strategy relies on being able to fix nuclei in order to do these. So this is all single nucleus RNA-seq, and that's sort of easier to do with our way with SkyRNA-seq than it is with other tools. So that's one reason. And then the second reason was, why not kind of a targeted approach where we're reading out say 100 genes instead of the whole transcriptome? And I think that there's a strong argument for doing that if you're looking, if you're studying the genes that govern one lineage and you really have highly reliable markers that you can kind of interrogate. That might be great because it would dramatically cut down on the sequencing cost of the experiment, and you might be able to kind of ignore lots of cells from tissues that you don't expect to be affected and whatnot. And I think in the future that'd be a great strategy, but since this is the first time we're doing this, we want to just look at everything.
Thanks. Nancy, you're muted.
There's more questions, but I think we could put those in the panel and check there. Let's move on to our next speaker, Bo Wang from Stanford Bioengineering, talking about single-cell atlas mapping through metazoa reveals patterns in cell type evolution.
Thanks for the introduction and the opportunity to speak. And as we all know, cells are the basic unit of life. Now, how do they evolve in animals? So with single-cell atlases from almost every corner of the tree of life now, it has just become possible to approach this question. So the idea is to map cell types in organisms across long evolutionary distances. But the challenge here is that the complex evolutionary history of genes can blur cell type relationships. So let's first consider a conceptual scenario. An ancestral cell type is defined by its co-regulatory complex, controlled by some transcription factors upstream of a series of target genes, which form the functional machinery of the cell type. The cell type then diversifies into two daughter types, each now adding a new transcription factor into its co-regulatory complex. So this leads to the addition of new downstream functional genes and the replacement of existing parts. And as evolution continues, some genes might be lost, while others might expand to have duplicated paralogs. This causes parts of the gene regulatory network to be substituted by new paralogs, which further reduces the similarity between cell types. To solve this problem, so we have recently developed a cross-species cell atlas mapping method, which we refer as SAMMAP, self-assembling manifolds mapping. So here's the idea. Consider species 1 and 2. So we begin with a gene-gene graph connecting homologs between the two species. Unlike other single-cell data integration algorithms that rely on presumed one-to-one gene orthology, SAMMAP allows one-to-many homologs to account for gene duplication events. We then translate each species' features into those of its mapping partner to map the atlas manifolds, which we constructed by SAM. So SAM is a robust manifold reconstruction algorithm that we developed earlier. And based on the mapped manifolds, we then compute the spatial correlations of the gene expression for all homologous gene pairs and then reweight the homology graph using the correlations. So this cycle is repeated for a few times to gradually refine the gene homology and prune the many-to-many homologies to only account for ones that are expressed in the same mapped cell types. So to verify the method, we first apply SAMMAP to the frog and the zebrafish embryonic atlases collected in the Klein lab a few years ago. And these data sets have matched ontogeny, which can be used as a reference of comparison. So as shown here, SAMMAP aligns the two atlases while maintaining a high resolution for distinguishing cell types in each species, with a few example mapped cell types highlighted in circles. And SAMMAP links correctly 26 out of 27 expected cell type pairs based on previous annotations, with the thickness of the edges quantifying transcriptomic similarity. And inversely, SAMMAP does not link cells that are not developmentally related in most cases, suggesting a high degree of agreement between transcriptomic similarity and ontogeny relationships. A few notable exceptions, though, are secretory cell types that do share similar functions and common gene modules, which are highlighted by the black edges here. So these exceptions support the notion that cell types can be transcriptionally and evolutionarily related, despite having different development origins and even arising from different germ layers. I want to quickly mention that we have benchmarked SAMMAP against several broadly used single-cell data integration methods and found that they only give minimal alignment between the expected cell type pairs. We also compared the results when restricting SAMMAP to using only the one-to-one orthologs instead of the full homology graph. So even so, we identified similar, although weaker cell type mappings. So this suggests that at least for the frog and the fish comparison, SAMMAP performance is owed in large part to its robust atlas stitching approach. So the key benefit of using the full homology graph is to enable the genome-wide identification of paralogs that have greater similarity in expression across species than their corresponding orthologs. So SAMMAP identifies hundreds of such events with two examples shown here. So there's a minimal overlap between the orthologs pairs. In fact, in these examples, either zebrafish copy or the frog copy is only expressed. Strikingly, the missing orthologs are then replaced by one of their paralogs. For example, zebrafish SOX8A is substituted by SOX10, which has overlapping expression in neural crest cells with frog SOX8 with the co-expression highlighted in green here. And these substitution events can arise as the result of gene duplication followed by diversification of paralogs. In addition, genetic compensation by transcriptional adaptation, where loss of function mutations are balanced by upregulation of related genes, can also give rise to this type of signatures. And these results illustrate the potential in leveraging single-cell expression data to link molecular evolution to cell type evolution. So far, I have introduced SAMMAP, which explicitly comes for the complex evolutionary changes for gene families during cell type mapping. And in collaboration with many research labs, we have so far successfully mapped more than a dozen species. The mapping results appear to be highly consistent to expert manual matching, and in many cases help the cell type annotation, especially in non-model organisms. But we have also observed, of course, limitations, especially when comparing species with extremely long evolutionary distances and completely different body plans. So these limitations may be attributed to the assumption that there should exist one-to-one correspondence, orthology of cell types between organisms. But this is only true if cell type diversification happens before speciation. In contrast, if cell type diversification happens after speciation, the diversification process should follow independent trajectories in two organisms. So in this case, one-to-one cell type correspondence may not exist, and instead we expect to observe families of related cell types sharing common gene expression programs that originated from the common ancestral state. So to test this, we mapped vertebrate embryonic data sets against two flatworms that my lab studies as a bilateral sister group of vertebrates, we also included two non-bilateral animals, hydra and a freshwater sponge, to sample a broader range of metazoan groups. So we performed all 21 pairwise mappings between the 7 species, connecting about 1,000 annotated cell types. So among all these mappings, we found neural and muscle-like cell types to show dense many-to-many connections among them, supporting the notion that ancestral cell types may have diversified into families over long evolutionary distances. To quantify the connectivity of the cell type network, we use transitivity, in which two mapping partners also link to at least one common cell type in a third species, forming triangles, with an example highlighted in red here. So more triangles does an edge belong to, better supported is that mapped cell type pair, and more densely connected is that cell type to its family. And indeed, neurons and muscles have high connectivity compared to the overall cell type graph, and all mapped pairs share a large number of genes, suggesting that the cell type families are defined by deeply conserved gene expression programs. And taking muscle cell type family as an example, so this includes not only muscle cells in all bilateral animals, but also sponge and hydra cell types that are non-conventional muscle cells, but have been implicated to drive animal contractions. So SciMap identified broad transcriptomic similarities between all the cells, including genes from orthology groups spanning diverse functions, actin regulation, cell adhesion and stability, and signaling. I want to draw your attention to several transcriptional regulators shared among the cells. So previously, known core regulators involved in myocyte specifications such as MyLD, E12, and MAF2 are enriched only in bilateral and vertebrate muscle cells. Instead, we found homologs of muscle link protein, CSRP, and a FOX group 1 enriched in muscle cells from all seven species, indicating that the transcriptional regulation of muscle-specific gene expression may have evolved earlier than previously appreciated, and rely on a set of regulators that are different from what are typically assumed. So in particular, the majority of the muscle cell-specific FOX proteins are FOXC orthologs, which have been implicated to regulate cardiac muscle identity in vertebrates. In our analysis, they seem to be also conserved broadly among animals except the sponge. And notably, FOXC is substituted in subsets of muscle cells by FOXL and FOXG. And the FOXG is particularly interesting as it is enriched in 3 of the 4 invertebrates, suggesting that FOXG may play an underappreciated role in muscle cell specification outside vertebrates. So taken together, by mapping single-cell transcriptomic analysis, we identify hierarchical patterns in cell type diversification. We observe one-to-one cell type mapping between animals that are relatively close, typically in the same phylum, whereas among animals over long evolutionary distances, we observe interconnected cell types forming distinct families which are defined by deeply conserved regulators. And I believe this is just the beginning, and a lot more discoveries are yet to come, with more and more data sets becoming available to better sample the diversity of cell types in the tree. And this work is led by Alek Tereshansky, a phenomenal graduate student, and our work has benefited hugely from collaborations with Dell Aaron's lab at EMBL and Steve Quick's labs at Stanford. So with that, thank you for your attention, and I'm happy to answer your questions.
Thanks, Bo. That was very, very interesting, and it's a really elegant method. I have a question about, under this framework you've developed, can you identify cases of concurrent evolution of cell types? What would you be looking for, and how would that be possible under this setting? So
for the concurrent evolution, you basically would expect sort of 2 cell types that sort of evolve in parallel. That's what you mean in this context.
Right.
Yeah,
right.
so,
Getting to the same cell type with a cell type from two different lineages or two different evolutionary branches, right?
That's right. So I think this is a great question. So, in this cases, you would like to compare cells that sort of in the embryonic development or in a sort of a constant homeostatic turnover that you would capture a lot of transient cell types. Now you could map the sort of terminal differential cells and the transient cells. And we do see parallel trajectories, like for example, when we compare the zebrafish and the frog data, because they have the cell types at different time points along the development, and we do see parallel developmental timing and the cell types in that case.
Thanks, Bo. Let's take one last question from Juan Lugo. Impressive work. Much of the studies are always considering large scales in evolution. What would be your predictions on smaller scales, like a group of sister species or even in phenotypically and genetically divergent populations of the same species?
That's a great question. So, as I mentioned, we observe two types of patterns in the cell type evolution. So within the animals that are relatively close to each other, we actually see a lot of homologies between cell types that we can draw one-to-one correspondence. And between animals that are very separate along the evolution, actually, you only observe cell type families which share ancestral gene modules. So what we really want to do is that we have data sets to cover both of these evolutionary scales. And with the animal that are close to each other, we can really construct the phylogenetic tree of cell types. While, when you have animals that over long distances, you might sort of track how the cell diversity that are built in different branches of the tree of life. So one of the focus that our lab is focused on, because we work on flatworms, so we want to sample as many different flatworm species as possible so that we can reconstruct the trajectories of evolution of each cell types. I think the challenge here is that, when people are trying to decide which species to sequence to construct a cell atlas, they try to sort of distribute that effort as much as possible, right? Like they don't want to sample a branch of evolution repeatedly by sampling close related species. But I think this is going to come when we have a relatively good coverage of the tree of life, and then we're going to have data that concentrate on a specific branch of the tree.
Thank you very much, Bo Wang, for this excellent talk. And we'll move now to our last talk of this day. And we'll have Noah Greenwald from Stanford University, who's going to tell us about accurate whole cell segmentation of multiplex imaging data by combining large-scale data annotation and deep learning. Thank you, Noah.
Great, thanks. I'm really excited for the opportunity to share with you all our new algorithm for accurate segmentation of cell imaging data. There's now a number of different available multiplex protein imaging platforms. The underlying technology for each of these is a little bit different, but the output is essentially the same, which is a high-dimensional image where each channel in that image corresponds to one of the proteins that is being acquired. This data is really useful because it allows us to look at the co-expression of these different proteins within the individual cells in the tissue that we've imaged. In an analogous way to how single-cell RNA sequencing or CyTOF data might be analyzed, we can look at the patterns of co-expression of these protein markers in order to understand what the cell types are in this imaging data. However, the really exciting thing is we can take that a step further, and because we have the location of each of those cells in the original tissue that we image, we can map back those specific cell phenotypes or cell subtypes to the original sample. For example, we can see how cells of a certain subtype tend to cluster or are spatially located near those cells of that same type or cells of different type. This is really powerful and is a great lens to looking at either healthy or diseased samples across a range of different conditions. However, these types of analyses cannot be performed directly on the image data that comes off of the instrument. These images are essentially just matrices of pixels where each pixel in the image corresponds to the expression value of the specific channel that you're looking at. In order to understand what's going on at the level of a cell, it's necessary to first define where each cell is located in the image. This process is known as cell segmentation, and in tissue, it turns out to be a fairly complex challenge and has been a major barrier to analyzing these imaging data sets. The reason this is challenging is because tissue data is quite complex. These cells are very densely packed next to one another. There's often a lot of heterogeneity in cell shape and cell size, even for cells that directly adjoin one another. And there's a large degree of variation across different imaging platforms, different tissue types, and even different experimental conditions. To address this issue, we created a novel deep learning architecture, which we're calling MESMER. MESMER is specifically designed for segmenting tissue imaging data. The inputs to the MESMER model is a two-channel image. The first channel, which defines the membrane or cytoplasm of the cell, is used for cell shape. The second channel is a nuclear marker, which defines the nucleus of the cell. We pass these two channels to a normalization function and then directly into the deep learning model. What the deep learning model does is predict both the center and the shape of each cell and each nucleus in the image. We then take these predictions and post-process them to create the final segmentation output, which is a mask representing the shape of each cell and each nucleus in the image. As you may be aware, deep learning models require large amounts of training data in order to get accurate performance. We created TissueNet, which is the largest data set to date of cell segmentation, training data for cell segmentation. TissueNet contains more than 1 million paired annotations for both nuclear and whole cell data. If you're interested in the process that underwent the creation of TissueNet, you can check out our preprint, which I've listed here below. One of the most important considerations when constructing a training data set is that it has sufficient heterogeneity to represent the range of different examples you want models trained on that data set to handle well. To this end, we made sure to include images from a range of different tissue types so that models that are trained on TissueNet would work well across a range of different tissues. We used TissueNet to train the MESMER model and then benchmarked it on a held-out portion of the data set. Shown here is the accuracy as assessed by the F1 score, where an F1 score of 0 would indicate no agreement with the ground truth, and an F1 score of 1 indicates perfect agreement with the ground truth. What you can see here is that MESMER is substantially more accurate than previously published cell segmentation approaches. To demonstrate this a bit more visually, we generated ground truth segmentations for colorectal carcinoma. What we've done is color each cell by the ratio of the size of the predicted cell to its matching ground truth cell. So for example, in this true segmentation data, everything is colored gray, corresponding to this 0 log2 fold change between the predicted and true size, and the F1 score is 1. If we look at this same image and the segmentations generated by MESMER, we see overall that many of the cells are colored this middle gray area, indicating we're doing a good job capturing the size of these cells, but there are rare cells on the extreme ends of this log2 spectrum. When we compare that to the previously published models, you can see a dramatic difference with a large increase in the number of extreme colored cells on both ends of the spectrum, as well as cells that are completely missed by the methods, which is a major limitation if you're analyzing tissue imaging data. One of the benefits of TissueNet having such a heterogeneous collection of tissues and image platforms is that models trained on it are really generalizable. Shown here is example output from MESMER on 4 different tissue types and 4 different imaging platforms. The important thing to note here is that this is the exact same version of the model with the exact same post-processing parameters and no manual tweaking required for it to work across this range of different tissues, which is currently a major downside, I would say, of many segmentation approaches, that they require a lot of fine-tuning. But because TissueNet is so heterogeneous, we can train a single model that works out of the box on this diverse set of data. We next wanted to evaluate how MESMER compares to human annotators. We enlisted a number of pathologists to perform a blinded evaluation where we showed them 2 segmentations of the same image, one generated by the algorithm, one generated by humans, and asked them to evaluate in a blinded fashion which was more accurate. What you can see here is that these ratings, while there's some variability, are all clustered right around 0, indicating that they had no preference between the model's annotations and the human annotations. When we break this down by tissue type, we see some differences. For example, immune tissue, there was a slight preference for the model. The colon tissue, there was a slight preference for the annotators, but overall a similar pattern where these annotations are all clustered around this 0 axis, indicating that we've actually achieved human-level performance for this task, which is really exciting. Now that we have this accurate segmentation, I wanted to highlight some of the cool applications that we can use this for. As I mentioned before, Mesmer generates segmentation predictions not only for each cell, but each nucleus within each cell. What this allows us to do is look at the subcellular localization of imaging markers. So on the top here is a number of different proteins that have known subcellular localization patterns, and on the bottom is the corresponding segmentation predictions for both the nucleus and cell of each cell. What we can do is use this to compute the subcellular localization of each protein marker, and then do that same calculation for the ground truth data for both the nucleus and each cell. When we compare these to each other, we see that there's excellent agreement between the subcellular localization prediction of each of these proteins from the generated from Mesmer's segmentation as well as from the ground truth segmentation. Another common task is quantifying cell subtypes. To evaluate how Mesmer impacts this task, we generated segmentation predictions from a data set including 3 phenotyping markers, CD8 for T cells, CD14 for monocytes, and keratin for tumor cells. We generated segmentations via Mesmer and also from our ground truth data from the annotators. We then used the same gating scheme to classify each cell into one of 4 potential subtypes. What you can see here is that in each image, the proportion of each cell in each cell subtype in the ground truth data agrees quite closely with the proportion of that cell from Mesmer, which is really important sanity check when using these methods to evaluate something complex, such as the different cell types that are present in the TME. Finally, we wanted to make this easy to access, so we've created a number of different ways for users to run the model on their own data. We have cloud-based approaches such as the deepcell.org website, plugins for ImageJ and QPath, 2 popular analysis software, but we also have command line tools such as a Docker image and a Jupyter notebook so that a number of different users can access the model in sort of whatever way works best for them. All of the details are in the preprint, which I mentioned below. And you can also check out the tutorials on our GitHubs. Finally, I wanted to thank everyone who was involved with this project. This was a really great collaboration with Dave's group at Caltech. Especially wanted to thank Geneva, my co-author on this study, both of my advisors, Mike and Christina, as well as Liat, who now has her own group at the Weitzman. Thank you for your time, and I'd be happy to take any questions.
No, there's one question here from Sweta Murali. How well does Mesmer perform in multilayered tissues? For example, if there are cells partially obscured by cells from above.
Yeah, so I would say it's not superhuman. It was trained using human annotation. So if you, as a relative expert, can look at the tissue and get a sense for what a accurate segmentation should be, the model is going to get about that level of performance. For tissue types where it's just so crazy and even a human looking at it isn't really sure what to do, the model is not going to be able to sort of solve that problem for you.
Thanks.
Another question, does Mesmer work for 3D images, such as 3D confocal images?
Not yet. So if you have Z stacks of images, right now what you can do is run Mesmer sort of serially on each of those slices, and then you can stitch the predictions back together. In order for a model to understand how to do 3D segmentation, we would need 3D training data. And that, as you can imagine, is very laborious to create. So it's on the to-do list, but currently the model can be run on each of the 2D slices independently, and then you can stitch them together to get 3D segmentations.
Great. There's also a question here from Joanna Petrescu. It's very impressive how well your tool works on multiple tissue types. Have you tried it for segmenting cell types with more elaborate morphologies, such as neural tissues, neurons?
Yeah, so you may have noticed that there was no brain data in this TissueNet dataset, and that's because it really is such a fundamentally different problem. I think that's one area where 3D might actually be easier rather than harder. When you take a 2D slice of the brain, it's often really hard to figure out what axons are going to what neurons and how these things should be connected, because you're really cutting through what's sort of like a ball of spaghetti. And it's only if you had a more 3D structure that you would even be able to understand how to link those together. So right now in the brain, we're saying we can do nuclear segmentation, and anything more than that, I think, is going to be require more sophisticated approaches.
Thank you.
Do we have time for one more question? So there's one question is from Di Hu. How does Mesmer perform when the membrane signal is not fully clear? For example, in some live imaging data?
Yeah, that's a great question. And also, for example, what if you have cells that just don't express the membrane marker that you've imaged? This is a common problem. And everyone has been on the quest for the perfect membrane marker. I think we can now definitively say it doesn't exist. And so you just have to bake that into the algorithm. What the algorithm does is when it's unclear, exactly what the shape of the whole cell should be, based on that membrane marker, it will default to just doing nuclear segmentation, so that if you, for example, have an epithelial marker as your membrane marker, and there's immune cells that are in your image, it's not going to just skip them. It will just segment the nucleus in the cases where it's not clear what the shape of the whole cell should be. For those cells that do have robust, strong staining of a membrane marker, it will perform whole cell segmentation on those.
Awesome. Thank you very much, Noah.